of large-scale models and shaped community. So Marcus, you can start. I think we can hear you well.
Yeah, Antonio, do you hear me?
Yeah, we hear you.
Yeah. Thank you very much. I'm going to share my screen.
And here, who can see it?
Yeah. You can see it?
Yeah, who can see it?
Okay, that's good. Let me see, I'm trying to optimize this a bit. You still see it? Antonio?
Yes, we see it. Yeah.
Mm-hmm. Okay, so I cannot see you now while I'm giving my presentation, so please just shout up if you if it's not working or something is not clear. Thanks a lot for the opportunity to talk here. It's very nice to have this view in this into this seminar room that I know so well. So in order to have a background for the story I'm going to tell you, it's of interest to look at this timeline, which sort of describes my journey with NeuroMat, and you will see how this is interrelated with the scientific work. So I first came into contact with the people around NeuroMat, I guess latest in 2012, when we contributed to LASCON 4. Of course, at this point, the LASCON was already very well established. And then a year later, NeuroMat was founded. And again, a year later, my lab published a model of the cortical microcircuit that's now called PD14. And much of the talk here will be about the history of this. In 2017, I became a member of the International Advisory Board of NeuroMat. And already in the same year, there was a first independent reproduction of the PD14 model by people of NeuroMat. Renan Shimura, who is the first author here, and also Antonio Rocchi. And since then, the collaboration formed, and we had many visits. In 21, there was another reproduction of the model, now based on the neuron simulator by Romero et al. And finally, then in 25, we had a joint community publication looking back at the history of this model and its problems. So this model is a critique on the minimal model approach that we are working, that was so successful in physics. So the standard strategy here is that we have some phenomena of the brain, and we build a minimal model that explains this phenomena. And so then we end up with n models that are each describing the mechanism that is underlying the phenomena. But this, of course, then raises the question, are these models compatible? And how can we combine them to build models of larger explanatory scope that explain us higher brain functions? There seem to be no clear pathway. And if we have this situation, then we basically violate a fundamental principle of the scientific method, where one generation of scientists can build on the ones before, as indicated by this quote here from Isaac Newton. So what I envisage is a strategy where we incrementally have models that are building on each other, and where we can iteratively include more and more mechanisms so that we come to a more complete understanding of what the brain is doing. And a part of this is when we are starting to talk about more complex model is that we need to turn to simulation, because not in all cases we can solve these models analytically. By now, I think it's fair to say that in computational neuroscience, simulation has established itself as a third pillar next to experiment and theory. My dear colleague Sonja, who some of you know, once said, never trust a theory that has not been verified by simulation. And you can understand this in different ways. One is, of course, it's fine to use simulation to check whether you got all minus signs correct in your theory. But direct simulations can also validate a theory by showing that the sequence of approximations that you necessarily do still leads to qualitatively meaningful predictions. But still, the real power of simulation, I think, is that you can go beyond what is accessible by analytical tools today. The case of computational neuroscience, for example, by replacing current-based synapses by conductance-based ones. Often when I review papers, I complain that the authors have just checked that their math is correct, instead of tackling by direct simulation a model that is a bit more complex than what they capture by the theory. It seems almost that authors are afraid of differences between theory and simulation. So I think I'm mentioning this here because I think that the combination of theory and simulation was very important for the success of NeuroMat. And in NeuroMat's case, this simulation pillar was very much implemented by the NeuroMat's SimLab that was headed by Antonio Rocchi. So in this last, let's say, decade or so, our formal framework in computational neuroscience has, with respect to simulation, has advanced a lot. And this progress is documented here in this community paper from 2019, where the researchers described that we have now reached a situation where we can formally describe neuronal network models independent from a particular simulation engine. This is not trivial. It's not true for all branches of science. For example, if you think about climate research, there basically the model and the simulation code is one thing. It's very hard to separate this. But the fact that we have made this separation brings us a lot of advantages. For example, we can run many different mathematical models on the same simulation engine. And that, of course, enables us to validate and optimize such a simulation code and operate it as a scientific infrastructure. And in addition, there are some models that are simple enough so that we can simulate them on different simulation engines. In this example here, on the nest simulation code, the neuron simulation code, and even the Spinnaker neuromorphic hardware. And this is also a big advantage because it enables us to cross-validate these engines on the highest level. So, of course, in the past, we have studied the brain because we want to understand brain function. This is the main motivation behind computational neuroscience. But through the rise of AI, suddenly we have gotten a responsibility that may be far more relevant for society. And this is depicted here on the slide in the lower left here. So the modern AI algorithms that we have are optimized for present day computers with their strong CPUs and GPUs, but we are paying a terrible price for this. Our brain consumes about 20 watts, whether a supercomputer consumes 2 megawatts, or for example, in the recent edition of a supercomputer at our site in Jülich, it's even 20 megawatts. So meaning there are 6 orders of magnitude in between. And also we are reaching the end of Moore's law, where the structures of our silicon becomes smaller and smaller, and thereby also the switching times. That means we cannot overcome this by further miniaturization. I will come back to this. But what's really required is that we need novel computers based on principles of the brain to overcome this. And here is a figure that shows how dramatic the situation is. The green curve shows you the energy that is consumed in the world by computation, and on the horizontal axis you have the years, and you see that there is this increase, and the yellow line shows you the world's energy production. And that means if the energy consumption of compute would grow like predicted, then in 2040 we would use all the energy that is produced in the world just for compute. But of course this is not going to happen, because we need electricity also for other important things like lighting, heating water, and so on. And so as a consequence, this curve of progress in compute and AI will flatten out, like shown by this dashed green line. So, and also, I mean, way before this, we see this already now. The use of AI becomes undemocratic and in the hands of a few people who can afford this. So this is an important issue, and raises the question: is there any chance that we can overcome this, that we can build computers that work more like the brain? And the two photographs on the right side should convince you that this is not hopeless. On the upper right, you see a photograph of a neuron, the black structure, and here you see the scale bar of 40 micrometers. So the cell body here has a diameter of about 20 microns, and below this you see also a photograph. This time the length bar is 20 nanometer, and here you see the size of a modern transistor. So meaning that in our technology we can already produce structures that are 3 orders of magnitude smaller than the relevant structures in the brain. I don't want to say that that a transistor can do all the things that a neuron can do, but this gives you an impression of the scales. And also, we don't need to be afraid of the number of elements. So the number of neurons in the cortex of the human is about 10 to the power of 10. That seems to be a gigantic number, but the number of transistors in a modern microprocessor is also of the same order. So the dynamic elements are not the problem. The real problem is depicted here in this photograph on the right. Here, this has a different staining. Here, the black structures that you see are the axons, the outgoing structures of neurons. The white spots are not neurons, but blood vessels. And you see that we have the same length scale here, 20 micrometer. And it turns out that the volume of the cortex is completely filled with this dense mesh of cables. There are 3 kilometers of axons per cubic millimeter connected in 3D, and this is something that no technology yet can realize. But if you remember the last slide where I said that our electronics is 3 orders of magnitude smaller than the structures in the brain, then this means that it also has faster switching times, because the switching times essentially scale with the capacitance. So the smaller an element is, the faster it switches. And so we have 3 orders of magnitude room to play with, to use techniques of engineering like multiplexing and so on. So this does, technology-wise, this does not seem to be hopeless. So is there any chance that we can identify fundamental working principles of the brain? And here I present you 2 slides that should give us some hope and motivation. On the right side here you see cross-sections of mammalian brains. Here is the mouse, and here is the human. And these mammalian brains, they look all very similar in their outer structure, which we call the cortex. It seems that eventually evolution has hit on a precious principle in the local cortical volume, so that it was useful to build just more of it. So our brains have a volume that is 1,000-fold increased to that of a mouse, and you can appreciate this a bit more on this figure here on the right, where we have the different species on the horizontal axis, and we are looking here into the local network of a few millimeter from the surface of the brain into the depths. And vertically here we have different brain areas that are processing auditory information, visual information, or involved in motor planning. And it appears that there is a dual universality across species and across modalities that are processed. And it's fair to assume that the potential origins of the low energy consumption are the spatiotemporally sparse spiking interaction that is going on in these networks, and that the architecture that is resulting from evolution somehow is an extremely flexible and fast learner. Now, that motivated researchers, at least since the seminal paper by Douglas and Martin in 2004 on the canonical cortical circuit, to look at this local structure of the cortex that comprises roughly a cubic millimeter of brain tissue, to find out how it is constructed and what its dynamics is. Here's a list of studies over the years. So our study here in 2014 is not the first, and it was also not the last. It contributed a certain perspective. And in our case, the question that we asked mainly is, how much of the dynamics that we see just comes out of the structure of the network? Therefore, we divided the cortex into 4 cortical layers. We decided that in each layer there's an excitatory population and an inhibitory population, but we made these 2 cell types exactly identical. The only difference is that excitatory neurons make only excitatory connections, inhibitory neurons make only inhibitory connections. And then we wired them up by what we could extract it from 50 experimental papers on the detailed anatomy. And out of this came simulation results like shown here on the right, showing that neurons in different layers have different spike rates. as it is in nature, and that in particular the inhibitory, the red neurons, have systematically higher spike rates than the excitatory ones. All this was known from physiology, of course, but it was always attributed to the inhibitory neurons being special, like being smaller, having shorter time constants, or being more excitable. All this might be true, but in this model, this is just an emergent effect of the network structure. So our idea of this model at the time was that it could be a building block for further, more interesting functional studies. Therefore, we tried to document the model as best as we can with the method that we had at the time, and we published the model code openly at this site here. And the model had some success in the sense that that it was cited several times, and more importantly, and these are the blue graphs here, that there were several studies who used the model to build on it. So they were not only citing our paper, but really using the model for something more interesting. And two of these studies I want to point out because they were very important for the steps that would follow later. And this was the first reproduction with the simulator Brian 2 by Renan Shimura and others, guided by Antonio Rocchi. So they transported the formal model description, which was not so formal at the time, to a different simulation engine, and could demonstrate that they can achieve the same results after finding some unclear points in the documentation, of course, and in our model code, but with some personal interaction, but not too much. We could sort this out, and then they wrote this paper about this. And then, like 4 years later, the same was done by a different group of researchers, but still centered around NeuroMat for the simulator neuron. And in this time, there was almost none personal interaction required to get this to work. And so there was a solid basis now for these results. And in 2005, when that was kind of the 10th anniversary of the publication of the original model, a group of researchers gathered to create this review here, discussing what happened in this 10 years and where the model was successful and what were shortcomings that could give us guidance for future more interesting models. And this group came up with a categorization that there were several fields of impact. One was, of course, the neuroscientific insight, requiring, of course, this reproduction. Then that the model was used as a building block for larger models. It was used for the validation of mean field theories, for testing out strategies for model sharing, and a bit surprisingly, it was used as a neuromorphic benchmark where the neuroscientific results are not so relevant, but it was just looked at how long does it take to simulate this. And looking back, it certainly helped that at the time when the model was published, there was also already one simulation code available that was quite mature and enabled other researchers to build on this model just by installing the same simulation code and running the model again. Of course, this is not really a reproduction then, but it gives you access to the tools. And for many years, there are tutorials worldwide on the use of this simulation code. Systematically, this was done at the Lascaux School, but also at CNS and as it's upcoming and other places. So what limited the impact of this model? Initially, certainly that it required a compute cluster and several minutes for just simulating 1 second of biological time. Then, in addition, the network has no function. It's not performing some useful operation. You can just look at the fluctuations of the membrane potential and at the irregular firing patterns that may have been disappointing for some. And as I said in the beginning, it's not formulated in the minimal model spirit, but as what we now call a digital twin. And that seemed not to be attractive for some researchers. Sometimes till today, we hear our colleagues saying, all I'm interested in, I can do on my laptop. That was clearly not the case for this model. But also, this statement cannot really be serious, right? It does not seem to be philosophically sound. Imagine high energy physics had stopped with tabletop experiments, and also why should the interest of a researcher scale with the computer technology that consumer companies provide? So the workshop participants concluded that the specific model, the PD410, can now be simulated on a laptop and faster than real time on a single compute node. So some of the barriers are gone. But also that the systematic education that we have been doing at the International Advanced Computational Neuroscience courses may have changed the situation, because now the next generation of researchers is much more used to work with simulation codes and with these large models. That goes so far that when we give a tutorial, sometimes we observe a feeling of awkwardness in the young researchers if we ask them to install software on their local laptops, because they are already so used to use cloud technologies and basically do everything in the cloud. But the model can also be criticized on a neuroscientific level because it has severe shortcomings. For example, it is lacking all power in the power spectrum in the low frequencies, whereas in nature there is a lot of power, and this may be due to the fact that we are describing all the synapses that are there in this network among the neurons, but each neuron receives 50% of its synapses from outside sources. And so one can also say that the model is heavily under constraint. And the full network structure is depicted here, where the microcircuit is sitting in this cortical sheet. But if we zoom out a bit, we see that this sheet has a spatial organization. The probability of two neurons to be connected decays with distance. And if we zoom out more, we see that the whole cortex decomposes in the human into about 100 areas. But each area is connected only to 10 of them. So even on the highest level, there is room for an interesting recurrent network. So to make the model more self-consistent, we turned to a model of the macaque visual cortex, that has 32 areas, where we placed one of these microcircuits into each of the areas as a representant, but connected them with what we know about the long-range anatomical connectivity in the brain. This model contains about 10 to the power of 6 neurons. That was at the time small enough to do production simulations on HPC systems. And this model then explained a number of other dynamical features. I will not show all of them here, just pointing at the power spectrum, where we now have this high power at the low frequencies that we see in nature, and also the distribution of spike rates across neurons, is much more realistic. And if we compute the correlation of the activity between the 32 areas, we arrive at a matrix that already has a high similarity to what we would measure in functional connectivity in the fMRI. So it really explains some aspects of the resting state of a monkey cortex. And from this on, models have now gone into several directions to either respect the inhibitory cell types better, or to transfer this idea of building a multi-area model that still has no spatial structure to the human, or the other way, concentrating on one area but taking the spatial connectivity into account. So while this work was going on, also the first large-scale neuromorphic computers have been built. These are two examples. The BrainScales computer at the University of Heidelberg, which is an analog device, and the Spinnaker 1 neuromorphic system at Manchester University. And these the availability and of this model and the fact that it could be well reproduced then sparked a race in neuromorphic computing for ever shorter simulation times and less energy consumption. Here you see some of these studies. That was a bit unexpected, but it led to a number of papers in very different journals. Most of them are listed here, so that after some time we thought it's time to collect all these authors and contrast their results, and it turned out that over just a few years, the availability of such a de facto standard has brought down simulation times and energy consumption by 2 orders of magnitude. You can appreciate here by this factor that's called a real-time factor that tells you how much slower is my simulation compared to real time. So at the beginning we had values like 2 or 4 here, and now with the latest results, systems are 20 times faster than real time, and this psychologically important threshold of reaching real time was first made by the Spinnaker neuromorphic system in 2019. And now when you collect all these results, you can of course also plot them. So here you see how the real-time factor goes down over time. This may be not so interesting. A bit more interesting is already this plot here, showing this linear dependence between real-time factor and energy per synaptic event, that basically tells you whatever technology you use, the main gain in reducing energy is just by simulating faster. But the real interesting plot is here, where the real-time factor is plotted over process node. Process node is the modern term for the structure size, so the width of the wires of your transistors. And here you see that the data points for CPU-based technologies in orange and GPU-based technologies in pink all nicely fall on a line with a slope of 2 in this double logarithmic plot. And this comes from the fact that silicon technology is essentially 2D. So if you halve the structure size, it means that you can place 4 times more transistors on your defined area of silicon. And so if all the these studies lie on this line, then it means that the researchers have done their job correctly. They have gotten out of the system what you expect. Electrical engineers call this scaling. But then you see points off this line here, the blue and the green ones, and these are the intrinsically neuromorphic systems. So either the Spinnaker system or green are FPGA-based technologies. They also sort of are on a slope of 2, but on a shifted curve, and there's a factor of 100 between them. And this is important because in the community there's some agreement that we would need a factor of being 100 times faster than real time in order to study plasticity and learning seriously, because otherwise the waiting times would be just too long. And you can see here that with the conventional technologies, we will probably never achieve this, because that would require extremely low process nodes that don't exist. Whereas on the neuromorphic line here, we have a fair chance with existing technology to reach this level. So in order to follow this up, as part of this study, we have created a new website that is shown here that provides you with the model code, with reference data for checking that the results are correct. But it also collects publications that have used the model to create more of these data points that I showed you. And so there's a call for everybody who wants to participate in this competition for less energy consumption and faster times to contribute new data points. So in summary, large-scale neural networks models today have become research platforms, sometimes also called digital twins. A model of the cortical microcircuit had some impact and became a de facto standard in neuromorphic computing. The community brought down simulation time and energy consumption by 2 orders of magnitude. And this development required progress in 2 things: reproducibility of modeling results and the combination of theoretical and computational approaches. And NeuroMat greatly contributed to this by taking on the challenge early on and by systematically educating a new generation of experts in computational tools. And clearly, these efforts need to continue on the national level in Brazil and in international collaborations. Thank you very much.
Thank you, Marcus. I think you couldn't hear, but we clapped our hands here.
Okay.
For you.
I'm still trying to. I'm not seeing. Now I see you again.
So,
okay.
Any questions from the audience? So I have a question that probably Antonio Galvez would ask, and probably. So the PD14, the Potchens-Disman model, emphasized the anatomical connectivity. So the main aspect was the anatomical connectivity, and not very much the single neuron model. You use the standard leaf model for all neurons. And NeuroMat, on the other hand, has always emphasized that single neurons, and in particular the stochastic elements involved in single neuron behavior, are important.
Yes.
How do you think one could conciliate the two views?
Yeah. So in this model, we purposely fully made the single neuron model deterministic and as simple as possible. But clearly, the natural neurons have an element of stochasticity. There is noise on the membrane potential, and so there's no problem in building models with this type of stochasticity that makes the spiking probabilistic, and build them into into such a model and then see what the combined result is. So what one has to consider is the magnitude of the noise we are talking about. So in this network model, in this balanced excitatory inhibitory state, the membrane potential fluctuations are in the millivolt range, whereas the channel noise is in the, I think, in the range of a few microvolts. So I would expect that in this state, the stochasticity of the spiking is really dominated by the fluctuations in the network, by the recurrent activity, and not by the intrinsic noise. Yeah, but depending on the input and so on, of course, both can contribute in some way.
Yeah, but there might, I mean, from our experience in simulating the PD model with stochastic neurons, and I'm going to show this in my talk later on today, we found some small discrepancies between the original PD model, the deterministic one, and the stochastic one, especially in the high frequency range. Not in the low frequency, which is important, of course.
Yeah, that's very interesting.
We had a peak. I mean, the original model had a peak in the high frequency range. I know that you solved this later. I mean, you were able to understand the origin of this. But
you mean at these unnatural 300 hertz synchronization. That's a clear defect of the model. Yes.
Yeah. Yes. But in the stochastic model, interestingly, we didn't have this. So for some reason, the stochasticity of the single neuron model was able to eliminate this effect. Because
that's very interesting, Antonio.
And so actually, I showed this the Aachen when I had a meeting 2-3 years ago, and I'm glad that later you were able to eliminate this effect by changing the balance between inhibitory neurons and so on. But the stochasticity, then, I mean, at least this for me shows that it was able to show some mechanistic element that was not present in the original deterministic model. Yeah.
Yes, it's an artifact that comes about by oversimplification in our original model. Yeah. So good level. And also, you probably then have the proof that these very fast oscillations, that they don't exist in nature, right? Because we have stochasticity. And that was always a question that was not really able to answer, because it's very hard to record such high frequencies experimentally.
Yeah, you're right.
Yeah.
Any further questions? Any other question? If not, so let's thank Marcus again.
Thanks a lot.
Here. Okay, thank you. And we can move on with the next talk by Mauro Copelli, who is already there. Thank you, Marcus.
Thank you.
And Mauro, you can prepare. You can introduce your presentation now.
Okay, let me try to project my screen. Hold on just a minute. I do this. Can you please confirm whether you can see my screen, please?
You can see it,
yes.
So you can start at any moment.
Just 1 minute. Let me just hide this thing here. Okay, so is it working? Yeah? So I wanted to talk about today about brain criticality in cortical states, which is work that we did in the late 2010s and early 2020s. And in particular, I want to call your attention to, for this audience in NeuroMat, how a model was essential to guide our thinking and change the direction of our research.
Mauro, sorry, you're not in the presentation mode. We can see all your slides on the left.
Yeah, that's strange.
Yeah, you should enter presentation mode, then you probably...
I did. I wasn't in the presentation mode. So can you see?
Now yeah, we see. Yeah.
Is it okay now?
See the other slides.
You still see the other slides?
Yeah, we still see all the slides there.
You shouldn't. Hold on. Let me try to share my whole screen and see what happens. Let's see if this works better now. Is it okay?
And now we see only the slide you want to show. Yeah.
Okay, great, thank you. So, so we have this National Institute of Science Technology for Neurocomputation, which is in its 1st year, and we're joining efforts among all these 48 researchers. So let me tell you about neuronal avalanches and brain criticality, starting with experimental data from Dietmar Plan's lab at the NIH. So they recorded LFP potentials from slices of red cortex and observed this burst of activity in your right LFP peaks, and they just try to count the number of active electrodes defined by some Z-score measurements in between silences, which you observe in the top plot. So they define this as a neuronal avalanche. So it's a burst of activity in between silences, and they count the number of active electrodes in between silences and call this the size of an avalanche. And then went on to estimate discount the number of avalanche with each side and plotted the probability distribution of observing an avalanche of a given size. And what he obtained was very interesting. It was like a power law distribution. So this power law could be very well fit by this 3.5 exponents, and this raised the attention of many physicists in particular, because this is a fat tail distribution, which means that the mean size of an avalanche, typical size of an avalanche, mean plus or minus standard deviation, these are all meaningless questions, because this distribution is ruled by fluctuations. And then they understood this as a non-trivial scaling variance statistics, which raised the attention of many people, because this field, a conjecture which was somewhere in the literature, that of the critical brain hypothesis, the brain is a dynamical system operates near a critical point. So in the cartoon to the right, in between some order and some disorder characterized in some way, which was to be determined, and right at the critical point, in the red point there, you expect precisely the kind of power law statistics that you observe experimentally. So if the brain is critical, what is the phase transition? Well, the phase transition can be characterized by a number of critical exponents, and the standard model, so to speak, for neuronal avalanches in this literature became the so-called mean field hypercorrelation class of model, in which you have some, in the subcritical regime, not enough activation among neurons, then eventually activity dies out, and you have some kind of what is called an absorbing phase, and you have strong enough connections, your activity becomes self-sustained, and in between, you are on the verge of dying off and not dying off, and that's your critical point. Precisely there, for this kind of model, you expect with a phase recognition between absorbed and active phase, you expect this exponent to be 3/2 for the distribution of sizes, and 2 for the distribution of durations, which is also parallel distributed. So this was very well known in the physics community for over 100 years. And people identified, oh, so this could be a signature of criticality. And this model, this class of model kind of gained traction as for theoreticians trying to understand this phenomenon. However, things are not so simple. Experimentally, these exponents are not always close to 3/2 and 2. Take, for instance, this experimental third of visual cortex experiment done by Woody Shu and collaborators in 2015. And again, you have the local field potentials. You try to estimate avalanche sizes and durations, obtain the parallel fits, and look at the exponents. And what you have is actual a scatter plot of exponents. And so tau t equals 2, which is what expect for the mean field calculation model. It's something like here, and tau equals 3/2 is outside the plot. So this is my field calculation model for the data which you see in black. So it's not a very good model for exponents alone, right? And some other previous results also suggested an intriguing dependence of parallel distributions on the level of synchronization, and data I cannot show you now. So we went into this European sensitized rat data because this kind of experimental setup does show different levels of synchronization. So we could try to see whether it played a role or not in these criticality signatures. So here you're seeing the coefficient of variation, which is a standard deviation divided by the average of the population firing rate. So here on the left, you see that low values of CV, on the right have high values of CV corresponding respective to less synchronous and more synchronous activity, which in the urethane setup, the brain of the animal spontaneously travels between those two regimes, or I mean, the discontinuous of regimes spontaneously. And each anesthetized animal has its own history, its own time series of this coefficient variation of the population fire rate, estimated like every 10 seconds or so. So we went on, and for each different segments of values of CV, we estimate to try to calculate spike avalanches instead of local field potential avalanches, choosing appropriate bins, and then looking at the size and duration of avalanches and doing the probability distributions. And you see the probability distributions, they change depending on CV. Sometimes they agree with, they come close to the expected value from inferior calculation, sometimes they don't. But the main point is that the avalanche exponents change continuously with the CV values, this degree to which your network somehow synchronous. And in physics, you expect, if there is a phase transition, you expect a well-determined value of each exponent, and other continuous variable exponents. So we asked whether there was a criterion to choose which are the true exponents if you are changing all the time. And the answer from theory is that yes. So a more rigorous test of criticality from a theoretical point of view is that avalanche has a lifetime of parallel distributed with those tau and tau t exponents. Then average size should scale with a lifetime t with a third exponent here called 1/sigma nu z. And we also observed that in our data. And furthermore, if those three parallel laws are true, then if the system is really critical, you should expect the exponents not to be independent, but to obey a third scaling relation called the Crackling-Noise scaling relation. So this was brought to the neuroscience community in by Friedman and collaborators in 2012. And note that the two sides of this scaling relation depend on CV and can be evaluated independently in our data. So the left-hand side comes from this kind of plot, and the right-hand side comes from two different plots. So the question is the scaling relation satisfied as CV changes? So I'm going to show you a plot of the left-hand side as a function of CV and of the right-hand side as a function of CV. And turns out that the two line crosses, at some intermediate value of CV, and moreover, the residence time is maximum where the two lines cross. We also tested it for free moving mice. This is not our data, but data obtained from free available data, and some supplemental neanderthalized monkey. So now that we have a criterion to choose properly where the exponents are supposed to be valid because they satisfy four scaling relations, are they compatible with this theory, mean-field calculation? So if we look here at the spread of exponents that we obtain in our data, we see that for anesthetized rats, tau equals 1.5 as predicted by the model, but tau t is not, it's 1.7. For the freely moving mice, the exponents are different, and the conclusions are that therefore they are not mean-field calculation, which is the black point on the graph. So it seems to require a different model. So this was very nice. This was the experimental data suggesting to us that the model we were working with was wrong. And we have different exponents for anesthetized and freely moving animals. That should not be surprising, precise, I think. And even away from criticality, from the hash regions, tau and tau t still follow the same linear relationship for different setups. For ex vivo turtle is there, so everybody is there. We don't understand that result today. So that was very nice. The main point of the paper was that. And then Tawan Carvalho, a PhD student in our group, kind of challenged us. He thought, is that really so? There are so many steps going on in this analysis. I'm not sure I trust the conclusions which are drawn from this paper. So he went on to prove this hypothesis with a model. And this is what I wanted to bring you here, the importance of models to challenge some experimental results and to see to which extent we can interpret experimental results, animal results. So what he did is the following. He took a model which does belong to the mean-field population class. I'm going to show the model in the next slide. But the main point is he subjected the model to the same constraints as experimental data. So first one is severe subsampling. So we have a model with a large N, capital N, simulated neurons, but sample only small n neurons out of it to do the analysis. Like we do, this is exactly what happens when you insert some silicon probe or neuropixels probe into a piece of cortex, right? And then subjected the subsample data to the same analysis pipeline as experimental data. So you parse by CV value, you bin the data, you check exponents and scaling relations, right? So the model he used was a spiking model with some excitatory inhibitory balance. The model came to be known in our community as the Gerstner-Galvez-Loscherbach model, GGL model, because it was inspired by work developed by Antonio Galvez and Eva Loscherbach, and also had been developed by independent by Wolfgang Gerstner. So this is essentially a discrete time probabilistic, so stochastic integrating fire model, if you want, in which you have this V variable, which would be like the main potential, as a function of which we have a probability of firing. And G controls the relative inhibitory strength, and you can see that the density of active sites as a function of G is a decreasing function, then reaches 0 at a point which, given the simplicity of the model, can be calculated analytically. So we are in the opposite regime of as what Marcus showed us previously, in the sense that this is a very simple model for which calculations can still be done and match the experimental results for this all-to-all topology very well. So we have a ground truth model for which we know has a critical point, and we know it belongs to this universality class. You can do full sampling, so sample all the neurons and calculate avalanches for the subcritical, supercritical, and critical regimes, and obtain very nice exponents precisely as predicted by mean-field calculation model. Please note that you have like 3 or 4 orders of magnitude in these power laws as expected by theory. So this is nice. This is just to check that the model does belong to the mean field directed population universality class. Now, what Taiwan did, instead of full sampling, let's sample just like hundreds of them out of the 10 to the 5 neurons and see what happens. So instead of this, what we got from the model was very interesting. So first of all, what you see on the right-hand side is that, I'm not sure my mouse can be seen, it cannot, right? So for a fixed value of the coupling parameter G or the inhibitory parameter G, for 3 different values have the 3 different colors, and for each color you see you have a distribution of CV values. So this already pointing to the idea that CV, which previously, when looking at experimental data, we consider as a kind of control parameter, which we do not control, but possibly we will interpret it as a variable control parameter in the data, is not really a good control parameter. G is a true control parameter, and it leads to a variation width of CV values. And you can see that within this variation, the fluctuations of the model, especially when they are when the model is subsampled, gives rise to, like in the blue raster plot here, asynchronous results, or it can lead to synchronous results with very tiny variations of the control parameter. This is what is important, and gives you size distributions and the duration distributions, which more or less match what you see in experimental data. What the model does not reproduce is this complex time series of CV values that you see in the upper plot, in the upper left plot, but it does reproduce the distribution of CVs that you see in the experimental data, right? So what Taiwan then did is proceed with the next natural step. Look at these avalanches, parsing the data by CV as we did with experimental data. Now we do with the model, and see whether the model reproduces the data. So I'm showing you again the data results. I'm showing the plot again because this was from data obtained in our lab. In the previous experimental results, we had data from our collaborators in Portugal, and we were very happy to be able to reproduce the results with data now obtained in our lab. And you see here in the right plot, again, a spread of exponents which fail to be close to the mean field error percolation. I remind you, the model belongs to the mean field error percolation model, but due to all this pipeline in analysis, subsampling, and etc., the exponents get distorted by the procedure, by subsampling, by many factors, and fall into these straight lines. You see here the model below. So in the upper plot, you have experimental data, and the lower plot, you have the model results. So the model results fall nicely into this dashed line of exponents, which is similar to the experimental data, but only if it's close to the critical point. If it's like 3% off the critical parameter value, then it drives away from the line observed experimental data. When you go, you move forward and try to obtain the experimental result, the exponents which do satisfy the cracking noise relation, which is the crossing in the left plot here, they are compatible within error bars with the model, with the data results, experimental results. So this was very important to us because that means that if you take a mean field error percolation model, subject it to subsampling, plus CV parsing, plus binning, etc., so this can lead to apparent exponents which differ from the true ones. I insist, if you look at the right plot there, what you're seeing, the true exponent values is the black dot. Tau equals 1.5 and tau t equals 2. But that's not what we observe if you subsample and do all these procedures. You observe this spread of exponents, which are artificial deformations due to the procedures. Therefore, what we had seen in experimental data, we don't know how to interpret anymore. Maybe because the data is produced by this subsample model. So despite the distortion exponents, the correct noise scaling ratio is still satisfied. So this was a shock to us. The correct noise scale relation is very hard to satisfy. You have to follow 3 power laws, obtain the exponents, and then match the exponents of these 3 power laws. So we were very surprised to see that even though in the subsample model, the exponents were distorted, while distorted, they still satisfy the correct noise scale relation. But this is true only if the model is within 2 or 3% of its critical point. Therefore, this deformed exponents somehow still bear witness to a signature of criticality. Therefore, this reinforced something that was already lurking in this literature, which is the fact that spike avalanches suffer from this methodological vulnerability, in the sense that spike avalanches suffer from this subsample issue, and therefore are, to some extent, a weaker signature of criticality. Importantly, a narrow interval, the control parameter, yields a wide range of CV values of the population firing rate, and therefore challenged the interpretation we had done for experimental data. So that CV is actually a poor control parameter, is not a true control parameter like we first interpreted when we were looking at the data. And this completely reshapes how we look at the results. So essentially, when we first, so what we see in the model here is that for a minor variation, like 3% around the critical point of the control parameter, we have a wide variation of CV values. This wide variation of CV values is what was observed here in experimental data. So this Fontanella et al. Paper, again, I remind you, is experimental data in which we have residence time peaking at what we interpret then as a critical point, which was is intermediate CV value. But you have to consider that according to this interpretation, you are very often away from the critical point, right? If you interpret CV as a control parameter. So in the interpretation of the experimental data alone, this will be at criticality en passant, in passing, right? However, if now we were guided by the model and we interpret that this whole range of CV values is actually obtained by very small fluctuations around the critical points, this completely changes the scenario. This points to a different interpretation, namely that you're not at criticality en passant, but rather you are all the time near criticality. So this is a slight different interpretation, which has further implications. Again, very wild conjecture because it's a very simple model. This means that this highly synchronous and high asynchronous states that you obtain in the urethane data could actually be just fluctuations around this critical point. And of course, these first results were obtained from experimental data, and the ones below were obtained from a model. The question then raised by us was that how can we possibly probe this hypothesis experimentally, namely that you are all the time near criticality? In order to do that, we had to go away from these avalanche statistics and probe new methodological approaches to test experimental data. Unfortunately, I don't have time to do this today. I'm already running my 30 minutes. But for those interested, we did this in two very nice papers, one led by Daniel Castro, who is now in Jülich, and another by Gustavo Combrainha, two papers in PRX Life, in which we use this approach called the phenomenological renormalization group, and actually confirmed that the prediction from the model seemed to be correct. So I thought it was very interesting to bring these results to this community here of NeuroMat to show you that the value of the model. Usually people say, yeah, well, the experimental data is the gold standard. Well, yes, but the way you interpret experimental data is also very important, right? Experimental data in itself doesn't mean anything unless you do something with it, and you work with it and do draw conclusions from what you observe. And what the model did here was to reshape our conclusions based on what we're seeing in the model, and then change the way we're addressing experimental data. And also other models. We are now trying to use these methodological approaches. We use the experimental data for more sophisticated model, like the Potchens-Diesel model that Marcus Diesel mentioned previously. So I just want to thank people who collaborated in these efforts that I mentioned today here, and thank you for your attention. Thank you.
Thank you, Mauro. We also clapped our hands for you. Questions? Just a quick question. You already mentioned that at the end of your talk. It's a pity that apparently Marcus had to leave. I know he's traveling. He's away from ULIS now. But you mentioned that it's important to, to go into more sophisticated models. I remember trying to find, say, criticality in the original Potchens-Diesel model. That was, say, 10 years ago with Newton Camidi and other guys. Do you have any hint about why we couldn't satisfactorily find the criticality in such a anatomically detailed model in comparison to, say, to this simple one that you presented to us?
Yes. So there are a number of possibilities. The simplest one would be that there's no criticality in that model. That will be the the strongest conclusions. But if I remember correctly, we were trying to obtain the signatures of criticality that we're trying to obtain in those days were precisely via avalanche analysis, right? And avalanche analysis, as I've just presented, it has some very strong methodological vulnerabilities. So if I get a clear signal of criticality with avalanches, I may be happy about it. But if I don't, I'm not sure it's not there, or avalanche analysis just didn't give it to me. So these other methodologies that we are using now, so particularly the phenomenological organization group, and also maximum entropy analysis, which we're also using in our group, these both seem to be more robust. And in fact, we've been confirming this signature of criticality in most data sets that we're looking at, including large-scale data from fMRI, for instance. We're observing this thing there, and we are very eager to apply them to models, to more complicated models. On the one hand, one natural thing would be to compare whole brain models when observed under these methodologies, which is something that Victor Gissa could contribute to, because it has such large models. But also in the cortical microcircuits scale, because we also see those signatures there. So we have, for instance, these data from the Allen Institute with neuropixel recordings with head fixed mice performing a task. We see very clear signal of criticality, and it would be nice to see whether a model which wouldn't have to have that many Potchens-Diesel model inside of it reproduce it. So we are working on it right now, and we don't have the results yet, but I'm very curious about it.
Thank you, Mauro. Any other questions? If not, let's thank Mauro again, and Marcus, who already left.
Thank you.
Break, and then since Bill Litton had to cancel his talk, our next talk will be by Victor Gissa at 11. Okay. Obrigado, Mauro.
Thank you. Obrigado.
you know, they are the coffee.
Antonio.
Hi, how are
I
you?
see.
Surviving with the
I'm
hot
good.
heat wave there in Europe?
Yeah, we have over 40 degrees.
Yeah, here it's about 12 to 13 degrees, so it's cold for us. It's quite
Ah, la
pleasant.
la.
Yeah,
I would
so
prefer that.
yeah, yeah. Well, you could have come personally, but next time.
Thank you, but
Yeah,
at
yeah,
some
no,
point,
I
I
understand.
should stay at home also a
Yeah,
little bit.
yeah, of course. So while people are coming, I think we can start, no? Because we have to
Yeah.
schedule to keep. So
Who's coming?
so
Yeah.
could you please come? Could you please come inside? And yeah, so you can prepare your presentation to be given to us
I
at
see
any
people
moment.
in jackets. Yeah.
Yeah, people are coming, and some people may be attending virtually.
Yeah.
Ainda tem gente lá fora? Yeah, they are calling. Yeah.
Who is it actually who's there with you? Is it school participants again, or?
No, this is not a school. This is a workshop. So these are well, the director of NeuroMat, Professor Osvaldo Baffa, here.
Hello.
There's some visitor from Finland, Pantelis Liumis. I don't know whether you know him, and then some researchers as well. Daniel Freiman, Marcela Svark, from Argentina. They are here. Claudia Vargas is coming. Florencia, so the director of the forthcoming NeuroMat CEPIX, and students from different departments from the university.
Okay,
So
great.
it's not
Yeah.
really a large contingent. It's I think that's
No
about
worries.
all. Yeah. So yeah. So I think you can start
Okay.
your
Then
presentation. Yeah.
I'll do so. Yeah. I need I'll be with you in a second. I'll just need to.
I think you have to select your screen.
Yeah, that's what I did.
Yeah. Your window. Yeah. Mhm.
You should see
Yeah.
it, I guess.
Now we see it. Yeah. Yeah.
Excellent.
You just
Yeah.
have to
Thank
move
you.
into present. Yeah, great. Now we can see it.
And do you see my mouse? Also my
Yeah,
cursor?
we can see your mouse as well, and we can hear you as well. Yeah, so
Okay.
you can
Then
start at any moment.
Go ahead.
So it's so let me introduce you. It's our pleasure to have Victor Giza here, continuing this session, giving the third talk of today. Okay. Carry on, Victor.
Thank you for this lovely introduction, Antonio. Yeah, it's a my talk is entitled From Brain Maps to Brain.
Victor, I think you're frozen, and apparently it's from your side. Can you hear me? Yeah.
Have not advanced. I did not do anything yet.
Ah.
Do you see this slide here?
Yeah,
Yeah.
we see the slide. Yeah, yeah.
Yeah, I have not advanced yet. I was.
Oh, okay.
Yeah.
Okay.
But I want to share with you some of our thoughts and activities in the context of twins. Yeah, and I want to frame it here. I'm going to start with this quote here, and then I want to guide you through the twin perspective. At the moment, there is a lot of activity in terms of twins that is ongoing, in particular in the context of AI-based twins. So I want to position our thinking in this context. This here is a quote that goes back more than 20 years to a lecture of Sidney Brenner, who worked on genetics, molecular biology. Following, we're drowning in a sea of data and starving for knowledge. The biological sciences have exploded, largely through our unprecedented power to accumulate descriptive facts. We need to turn data into knowledge. and we need a framework to do it. And I really like this one a lot, and I use this quote a lot, typically to introduce a way of thinking that data is not knowledge. And this also in the context in which he has used it, but it's not that simple anymore, especially not nowadays anymore. Yeah. The question arises actually, what is knowledge in this particular context? And here, one could argue this is a quote of myself. It's true in this paper here by Fosek et al. However, it has been heavily inspired by thinking of mechanisms by the philosophers that has been published in Philosophical Transactions by Mahammer, Darden, and Craver. Yeah, and I took some of their definitions of what a mechanism is, and I actually rephrased it here in terms of what explanation means. An explanation understanding as generating knowledge, which involves revealing the productive relation between causal activities on the one hand side, linked to their constituent entities on the other hand. Yeah? Please sit on this one for a second and absorb it. Productive relationship between causal activities and constituent entities. So you have a system composed of its entities, state variables, and causal activities is our understanding of the underlying causes that actually predict the evolution over time. This is actually the maps pretty well on the definition of what a dynamical system is, going back to Vladimir Arnold, requesting that a dynamical system is defined by 3 ingredients. That is a notion of time, number 1, space of state variables, number 2, and the presentation of an evolution law. Whatever the evolution law is that predicts the evolution of a trajectory in the space spanned by the state variables. Yeah? And this evolution law and this formulation leading to knowledge, what we call today knowledge, can take different forms. I'm giving here you the example of a damped harmonic oscillator. Antonio, can you see my mouse here when I wiggle it? Yeah, this one here?
Yeah, we see it. Yeah. Mm-hmm.
Thank you. Damped harmonic oscillator, linear system, which is represented here. If it's overdamped, then we can actually say, okay, it can be described by a potential function. If it's undamped, you have a Hamiltonian system. So sometimes there are some generating function that allow to describe it in a different way. Generally, we have the structured flow that is operating in the state space that is prescribing the evolution towards, here in this particular case, a linearly stable spiral fixed point. But there are also other ways that are unambiguous. For instance, the solution to this equation you can describe in a completely closed form. Yeah. So where there is a set of parameters that remain to be fixed, gamma, omega, yeah, and phi. But if you fix those parameters that are linked also to the initial conditions, particularly phi, then you can actually plot every single trajectory, yeah, over time as time series. And if you map it out for all possible parameters, what you have here on the left-hand side, you have the entire range of possible solutions as a data set in appropriate time series. Yeah? So you have data on the left all the way to a formalization, but epistemologically, this is actually fully equivalent. You can actually generalize this. Yeah? And even though that sometimes the generating function may not exist, or we may not be able to find an explicit solution for the differential equation, it may be a differential integral equation with time delays, etc. But the whole logic holds that we have an epistemological continuum, yeah, ranging from, on the one hand side, the totality of all possible time series and all possible behaviors as captured by the time series, or in other words, data, yeah, all the way to if we had all mechanisms under control and represented in algorithmic form, that would be equivalent, and we could actually navigate this particular space here. Generally, this is never the case. We do not have the totality of all mechanisms in place when we're looking for a twin or to describe a phenomenon, or equally, when we are here more on the left-hand side of this epistemological continuum, we never have all the data possible, especially when we're talking about experimental data, empirical data. So what we are dealing with is actually along the vertical component, we are dealing with a loss of information. What I just described to you may be true when we are navigating in 100% knowledge of everything. Yeah, but actually, when we go down to the vacuum of knowledge down here, then what we have to position ourselves are actually with approximation of what is happening up there. So very often we use toy or generic mechanistic models in order to highlight some of the phenomena, and there is loads of loss of information in order to describe the full real system. If we have not access to all the data, then we're dealing with empirical knowledge, clinical experience. We're looking back at what we have seen. We've never seen everything. And this is relevant, yeah. Clinical experience, for instance, when you have seen many different cases, yeah, then you are able to make some predictions about the evolution and predict individual trajectories, yeah. But what you're not able to do is generate what we call counterfactuals. So behaviors predict trajectories that have never been observed. Yeah? It becomes more algorithmic, more formal, and this is important for today's era where we're dealing with generative AI, deep predictive networks, large language models, which are essentially everywhere entering, estimating statistical resemblance, statistical higher order momenta, and then translating this into conditional probabilities, predicting trajectories to some degree, and this is actually quite well. But today, where we are dealing with is in terms of digital twins. On the right-hand side of the spectrum, these are mechanistic twins, where we know some of the mechanisms, but they are data constrained. Yeah, this is the state of the art where we are. Yeah. And what we would love to do is combine it one way or another with some activities here on the left-hand side, because very often this part of knowledge that we are missing, yeah, is a different one than the knowledge that we are missing here. So the ideal situation would be to have hybrid digital twins, where we are working with AI-informed knowledge that we are integrating into the mechanistic twins that has the advantages of, on the one hand side, performing counterfactuals, and especially with activities linked to medicine. This is what we are mostly interested in. You can actually perform real precision medicine of individuals, because you never have the entire data set available for an individual, yeah, because it's impossible to obtain. So we are obliged in clinical applications to go into this direction. And what I want to show you is actually how this is being done. But with this introduction, what I wanted to show, I wanted to position your thinking to give you a chance to recognize what we are dealing with are essentially two things. Looking into twins, digital twins, brain twins. Number one, we are dealing in reality actually with workflows. The question is, how do we operate these twins? Yeah? Not how can we build the best model. That's not the point. We need to build workflows allowing us to operate these twins in real world situations, number one. And number two, what we are actually dealing with is not how we handle our knowledge. That's an important element, but people focusing on this, how can we build the perfect copy? Yeah. What's more important is actually how do we manage the ignorance? So from our knowledge, the upper part that is missing to get to the 100%. Yeah. And this is a very important aspect. We do this is I will walk you through. Model construction is on the one hand side, how do we build these twins? The workflow to operate them, counterfactuals being our capacity to build scenarios and predict trajectories that have been observed before. So let me walk through. Yeah? The virtual brain is a principal full brain simulator in the infrastructure for digital neuroscience. And is as follows. This here is actually not an AI-generated image. What this is actually a snapshot from what we are doing in eBrains, in particular using the virtual brain simulator, where we are actually working. So this is directly coming from the platform where we build full brain network models derived from an individual's brain imaging data, allowing to reconstruct the geometry on the one hand side, and which provides us, which is obtained from the MRI data. And what you see here is the topology that comes from the connectivity that is being reconstructed and extract from individual. And then it's linked to the appropriate mathematics. What it looks like is as follows. So we are not dealing with individual neurons. This part I'm going to do fast because I think you will have other lectures on this, in particular by Alain, but we're dealing with mean field equations. So these are neural mass models that are scaled up from populations of individual neurons using different types of statistics, nonlinear dynamics, typically mean field approaches that actually allow to reduce a very high dimensional dynamic space to a set of 2, 3, 4, 5 state variables that are collective state variables, and then describing the collective behavior typically of mean fields or variances thereof. Here you have an example that is coming from a simulation. You have a raster plot, multiple neurons plotted over time. So these are action potential firings, each dot. Compute the spike density, which
Hi, Victor. I think we lost you. Voltou? Are you back? No. No. Yeah.
Can you hear me?
Now we can hear you. Yeah, but we had a kind of freezing moment there where we lost you.
I'm at work actually. I'm not on the Wi-Fi. So on the Wi-Fi.
Yeah.
I'm sorry. I do my best. So it's
okay,
quite network here. I don't know why it's having a hiccup. Do you see the next slide that is entitled "The Mathematics of the Virtual Brain"?
Yeah, we see it. Yeah.
That's an equation that underlies the construction of a whole brain network. Most of this has been derived actually together with Hermann Haken, our late Hermann Haken, a pioneer in the field of synergetics on self-organization. The mathematics is as follows. You have a differential equation. The state variable is typically a field. Typically, it's a vector field. It can be higher dimensional. Location x, time t. Then you have the neural mass dynamics, nonlinear, the type of mean field equations likely Alain will speak about. Then you have a convolution kernel, x minus x prime, so invariant under translation in space. This is the intracranial connectivity. And then you have the connectome that you see here be presented that is not translationally invariant. It's not x minus x prime. It's highly specific for the human brain. We refer to this as the connectome. It's not translationally invariant, and there is a large variability from one individual to the other. This is what we are reconstructing. You have a projection from one area to the other, and the signal propagation undergoes the time delay, which is being communicated via conduction velocity v. This is the time delay that you see here. Something from the math side that is very important, the fact, if this here were a translationally invariant, then we could write the entire equation as a partial differential equation. This is impossible. And as a consequence, we have a partial differential equation with no time delay, because you can actually integrate this into the kernel And then you have a PD, and you can push all the causal dependencies into higher order derivatives. The fact that this is not translationally invariant makes this a functional differential equation once we map it onto individual modes. Yeah. And this here determines the connectivity with fixed time delays depending on the distance. That makes it complicated and difficult and challenging to deal with. But this is specific for the brain. I do not know of a physical system that has this particular organization. Summarizing following situation, we deal with a brain. If we want to go all the way to the sensors, meaning to the patient, we need to span it in three-dimensional physical space. We map it onto individual network nodes, as you see here. So we're dealing with network node dynamics. And then we connect it through the connectome. And then what is very important, we map from the sources upon the sensors. In epilepsy, for instance, in individual humans, drug-resistant epilepsy, we drill a hole and we insert these electrodes that typically have 10, 15, or 20 contacts. And we measure the activity in the volume of the brain, or EEG, MEG, or fMRI. That's the whole workflow. Let me give you a few numbers and a little impression of what it looks like. We can, with 200,000 nodes, yeah, I showed you only a few nodes. On the previous slide, I showed you 165 nodes. But today we can go, we use GPUs for the computation that allows us to simulate thousands of nodes that get us to the spatial scale of millimeters. Yeah, and we get very high resolution. Yeah, that allows us to do this type of simulations. Here you have a cross section through the human brain. This section here is the hippocampus, so that's the cortex. And then the cortex folds in and builds this little spiral, if you wish, and this is the so-called campus, the hippocampal body. And I map it here on the right-hand side. So these are the regions, CA1, CA3, CA4 of the hippocampus. And on the right-hand side, we increased in epileptiform activity. This starts oscillating and then propagating. We see this exactly this type of behavior empirically all the time. There are waves of activity moving forward, penetrating into the non-epileptiform areas and then slowing down. The slowing down is what you just saw here. This is a famous spike wave complex, and then the activity stops. Yeah, so this is a very high resolution. We can map the whole into the brain, in the entire brain, and then we can generate this type of behavior. This is a full patient reconstructed the way I told you. In blue, you see the center of mass of each individual brain area. And then spanned in 3D, and then the needles, the SEG, stuck in that you see them represented here on the left. This is coming from computer tomography, CT. We are reconstructing the coordinates and placing it in here. And then once the parameters are set, we can actually run simulations as you see here. So you have a seizure onset. The time series that are being plotted here are the ones that are being measured by the yellow needles. This is the SEG. They mimic a seizure as it propagates through this particular patient's brain here, yeah, done completely autonomously, yeah, after the parameters have been inferred and set. And what you see in red is the energy measured here specifically on the sources, which is a sliding window integrated over time, just to show you a little bit where it's located in the brain. That's the capacities that we have nowadays today. And what I want to do now is in the remaining minutes to give you a little bit of a feeling what can be done with this hands-on, because we need the workflows. I told you, positioned your thinking a little bit, a perspective in terms of AI versus mechanistic twin, showed you how we build it. Let me show you some applications. Yeah. In a real world scenario, in the clinic, we have patient data that enter. It's typically structural data, yeah, and functional data. Functional means time resolved data, EEG, SEEG, MRI, or CT. We integrate this and build the twin exactly as I just showed you. But then we need to set the parameters. And the parameters specifically are location dependent, number one, and then they are mechanistically dependent. They are the pathophysiological causes. What does that mean? In epilepsy, it's the epileptogenicity that is represented by more physiological imbalances, typically EI imbalance. Yeah? In multiple sclerosis, it could be that increased time delays, so the demyelination of the connecting fibers. This we implement, but once we feed in the functional data, we perform Bayesian inference and estimate the so-called posterior parameter distributions, probability distributions of the parameters that identifies, that is a second level of personalization, if you wish, and then provides us with information that goes to the clinician and informs decision making. Specifically for the case of epilepsy that we are discussing here, you have the situation of drug-resistant epilepsy. The following is the situation. 1% of the human population is epileptic or becomes epileptic at some point in the life. Mostly children, adolescents, young people, but you have also late age or stroke-induced or dementia-induced epilepsy. These things can happen. Yeah. We treat this with drugs. In 60% of the cases, we succeed. That means in 40% of the cases, we do not succeed. In order to provide relief to the patients, we need to perform other procedures. State-of-the-art procedure nowadays is epilepsy surgery, if possible. And that's what you see here. For this, we need to know what tissue to resect. Often, and when possible, we enter into the brain, we measure the activity there. And then we have this type of time series. Yeah? And based on this time series, we have to infer where is the epileptogenic zone, which will then become the target for removal. Noteworthy, there has been no significant improvement of surgery success rate in 40 to 50 years, yeah? Which means there is something that we really are not managing very well. So, once we build the workflow, or this is the way how we operate, we build the workflow as I described to you, and then we perform inference. In order to perform inference, you need to have some data feature that you wish to sample through the process. What you in our hands, it was the seizure envelope that was the most predictive. Yeah? We tried many different features. Many other labs tried many different ones. The seizure envelope provides us with some identifiability of the parameters. Here you see a seizure, pre-ictal spikes evolving, yeah, and then seizure onset very fast, yeah, and then slowing down towards the end. What I just described, you see here in the frequency time diagram. Pre-ictal spikes, then small amplitude by high frequency, slowing down, other harmonics occur until you have seizure offset. So the seizure, the envelope is essentially tracing out this part, as you see represented here. This would be the envelope going down. Here you have some other seizures where the envelope I plotted in red. The same type of dynamics, you can plot this following way. This is pre-ictal resting state. And then this point here, it jumps at a different part of the state space and starts discharging very, very fast until you get to the offset and back to the resting state. So if you want the geometry in the flow of seizures, the geometry of epilepsy is a spiral. Rest state, onset bifurcation, spiral, offset bifurcation, back to the resting state manifold here on the left-hand side. This we can sample, and when we take the seizure envelope, every seizure can be represented like this. Almost every seizure, the ones, and almost all of them undergo bifurcations. Projected down here in the flow, This is what the seizure envelope looks like for this seizure. Resting state, then it goes up, you're sampling, envelope, and back down. That's the part that we sample. And that provides us with parameters, yeah, for each individual brain region related to the brain excitability in this case. Follow me here to the middle of this picture. This is actually how we sample. The way we sample is we run millions of simulations, yeah, many different parameters. Well, I exaggerate, 100,000s of simulations, yeah, many different parameter realizations, and then we are using SBI for the expert simulation-based inference. We are training a network out of which we are then extracting the posterior parameter distributions. In blue is a real distribution. For MCMC, Monte Carlo Markov chain, when we sample, we are tracing it out pretty well. Sometimes we used minor shortcuts, maximum a posteriori, where we're going into the center of mass of this probability distribution. If you do a simple Gaussian approximation, this is what you get. Importantly, you see here it's actually a bimodal distribution. Yeah, so this one here corresponding to non-epileptogenic, and this one here corresponding to epileptogenic. Yeah, the surgery has been performed, and you see it represented here. This is post-surgical MRI. You see the black hole here? This is the tissue that has been removed. The estimation that has been made, plotted as a heat map you have here. Yeah, this is only one single area. When you're in the higher dimensional space, what it looks like, just two areas, one area, second area. And so you have actually multi parameters that is being communicated to and with clinicians. Yeah. We are doing this systematically, and this is how we communicate. We pass them on a report. We call this VEP, virtual epileptic patient, and identify the brains, and then we provide metrics. Typically, the clinicians want to have it as simple as possible. Metrics, R hat value, confidence, etc., that provide us with our degree of confidence we have that the estimation is correct. Yeah? When we compare this again against some data and against retrospectively, is what it looks like. Whatever statistical analysis you will want to perform, here I'm plotting precision, you can do AUC curves, etc. They provide kind of similar converging results. This is a surgery that has been performed, compared against the clinical hypothesis. This is what comes from the virtual brain for seizure-free patients. Yeah? For not seizure-free patients where the surgery failed, you see not much change for the clinical hypothesis by construction. Yeah, but of course, yeah. But for the VEP, it falls down significantly. Why is that? Yeah, and we went into the data, multiple cohorts that have been analyzed. Yeah, so citations you have down here. What is happening? I invite you to think of the following: enter in to the brain, you get the. As a clinician, you analyze the data, scalp EEG, non-invasively. Yeah. You have the history of the patient, and then you where the the patient, and then you collect. Then typically by visual inspection, you assess the epileptogenic zone is located. Yeah? What we do with the virtual brain, full brain network approach, we perform a causal extension, not only to this space here, but to the entire brain space, yeah? Because we derive it from structural data, we equip it with mathematics, and then we model the forward solutions onto the SEG needles. Yeah? That allows us with the capacity to not only predict what is in the data space, but also outside of the data space. In other words, counterfactuals. Yeah? And that's exactly what is happening to a large degree. If you look at this plot here, if you look at the correspondence from the virtual brain prediction with regard to the surgery that has been performed, here for this cohort I just showed you, when it corresponds well, patients are seizure-free. But when it not corresponds well, it's actually down here to 40%. Yeah? Patients are not seizure-free, but the important aspect is the following. A third of all the cases that are failures, they fall into the case where the epileptogenic zone by the virtual brain has been identified to be in this regime here. Yeah? So this is being tested in a clinical trial right now. So this is the essence of counterfactuals. Yeah? And this could not have been done with a pure data-driven approach because you cannot step outside of the data space. We are testing this in a clinical trial that has been just finished across 13 French clinical centers where 356 patients prospectively underwent the virtualization and surgery. Their data were collected in these centers. We virtualized them in Marseille. We built the clinical report that I just showed you. Then we sent the data back for decision-making for half of the patients. For the other half, we did not do this. The other half served as a clinical, as a control group. So we have essentially a two-legged classic open trial randomized clinical trial. Yeah. So that's what I wanted to share here with you. I'm having an eye on the time, so allow me to stop here and just point out that everything I just described to you, you will find on eBrain's research infrastructure, which is Europe's digital neuroscience infrastructure. What I showed you in terms of digital twinning generalizes. So our most advanced case is epilepsy, where we are running clinical trials. We have another clinical trial that is in preparation for schizophrenia. This causal extension that I spoke about, please recognize it's not about extending it in space. Yeah? That was just intuitive here. It's an informational extension. Yeah? And that allows us, that actually justifies why it can be applied also to other applications. All the tools and workflows are applicable and accessible to you for free. Please simply register and have a look at the platform. And allow me to stop here. Thank all of my colleagues, TNG members in particular, and my friends and collaborators in particular here in the clinic. These 4 individuals have majorly contributed to this. So these are mid-level career researchers, and Hui Fang has done a lot in epilepsy. Marmaduke has done a lot in the inference, SPAS, and Pierpaolo on the modeling side. Thank you very much. I hope you're still here because I don't see you. Ah, I hear something. Good.
Hi, Victor. Hear the clapping hands?
Thank
We'll
you.
all clap hands for you. Thank you. Questions? Any questions for? Yeah, Daniel Freiman there has a question.
Hi, Victor. Very nice work. Thank you. Okay. I have just a question. If you study just a patient in resting state condition and you fit the model parameters, are you able to identify if this patient has or will have some seizure event or had in the past without using any epileptic information, I mean?
Okay. Are you talking specifically about epilepsy or general diseases? Yeah?
Epilepsy.
Epilepsy. Okay. We have a paper that came out last year in Nature Computational Science that does exactly what you're asking for. The patients have been, and 2 years ago in PLOS Computational Biology. What we are doing there is we are using interictal events. So resting state, but resting state of epilepsy patients may have interictal spikes. Yeah. So we are using resting state, but interictal spikes to estimate the parameters for the particular patient. And then what we are predicting is, we are estimating, inferring these parameters. Yeah? What we did in this in these two papers specifically is then predicting the capacity to stimulate it. Yeah, because you can also stimulate the brain of an individual subject, and depending on how the epileptogenic zone is organized, it reacts differently to the stimulation. You can trigger a seizure, stimulating with invasive electrodes. Yeah, so we use this as our validation paradigm. So we were able to use these interictal data to infer the parameters and then predict the responses to the stimulations. And this worked very well, way beyond the number of different surrogates. If you look this up, the estimation of the epileptogenic zone is not as good as when you do it with seizure data. Yeah, it's not as good, but the interictal activity provided us with a very good starting point. In the Nature Computational Science paper, what we showed is if you were, and that was a theoretical study, computational study, if you were able to trigger seizures with extracranial stimulation, such as temporal interference or some other form of stimulation, and trigger seizures and measure the activity with the EEG, you're actually able to estimate the epileptogenic zone to a very good degree, but you need to have the seizure activity. So once the brain stimulation technology advances sufficiently to do this non-invasively, we can use the entire workflow that I discussed to do a non-invasive estimation of the epileptogenic zone, almost at the same quality as with SEEG. And this will be breathtaking. So rest state is less informative, but if you cheat a little bit and make it a stimulation study, yeah, then we are getting some entry points. Yeah?
Very nice.
Thank you.
Thank you. Other questions? Well, we're running late, but let me ask a question. Since we're at NeuroMat closing event, and say one of the main efforts of NeuroMat has been in constructing stochastic single neuron models, as we all know, the Galvez, Locheba, and you're not using as an intrinsically stochastic, say, base model for your approach. You're using a mean field model. You can add noise. You can add noise to the model, but the model is not intrinsically stochastic. Would that be any advantage of introducing an intrinsically stochastic model from your initial equation, the one you showed us, instead of just having a deterministic one plus noise as you use in your approach? Or, I mean, this would, I mean, probably take, I mean, to the same kind of results.
Yeah. So we are not using single neuron models, stochastic Langevin equations of the single neuron. Yeah, this is true. We do use, in the equation that I showed you, there was a noise term in there. Yeah, so we do use stochastic equations on the field, on the neural field level. Careful, when I say neural field, I do not mean mean field equations, but I mean a field vector that has a spatial and temporal component, and it's this integral differential equation that we operate with noise. Noise plays a major role, especially in situations when we are dealing with non-epileptic activities. So we are extending much of the work that we are doing is applied to aging, or we are preparing 2 clinical trials in schizophrenia where we are able to predict medication. The metrics that we use here, I talked to you about the envelope functions for the seizures. I typically talk about these things, Antonio, because they are intuitive. You can see them with the naked eye, and it's most developed. There we have just finished the largest clinical trial worldwide ever performed with digital twins. So It's more advanced. The others are more in preparation. One of the biggest challenges is data features, yeah, because it's not that evident. So we are using higher order momenta of typically connectivity features, functional connectivity features, time evolving functional connectivity. So the data features we are using is we are sampling manifolds in the state space. We are trying to extract the manifolds on which the dynamics evolves. And these manifolds are being sampled and driven by noise. Noise plays a very important role because without the noise, we are stuck in several of these attractor spaces, but with the noise, we are actually exploring and sampling the manifold. And this has been demonstrated over and over again in the empirical literature that this is informative, also for cognitive performance, for instance. And we are finding the same in the model, the same type of behavior. So with all the metrics that we are using then to perform the inference, they require stochasticity. Otherwise, nothing works. So not on the single neuron level, but on the level of higher dimensional integral differential equations expressed in Langevin form and or in as Fokker-Planck equations. These are the mathematical tools that we're typically using. One last sentence, because it's the last one. The biggest mathematical challenges we get are, we have high dimensional equations, yeah, that have discrete time delays with translationally non-invariant connectivity kernels, yeah, in the presence of noise, yeah. Just numerical simulation of this is a big pain, yeah. And then the mathematical treatment, get all these elements together, it's very challenging. And as I said, this type of mathematics, the interaction kernel that is not translationally invariant, this is not what we know from physics, because all the forces are translationally invariant, yeah. In the brain imposes this type of structure upon us. Yeah.
Victor, perhaps this is a great problem for the next CEPIX, NeuroMat CEPIX, the next generation of NeuroMat. Okay, thank you again. Let's clap hands again for him.
Have fun. Yeah. Bye-bye.
Bye-bye, thank you. So let's move on. Now I think it's my turn, so I have to. Okay, so yeah. Okay, so it's last talk of this morning's session by me. So you already know me, so I'll give a kind of brief overview of NeuroMat SimLab contributions along this decade. So yeah, so the NeuroMat simulation laboratory, you call SimLab, it was established in more or less 10 years ago, 2016, there at the Department of Physics at the University of São Paulo in Ribeirão Preto, Faculdade de Filosofia, Ciências e Letras. Well, it's a dedicated high-performance computing infrastructure to enable NeuroMat neural network simulations. So that's the idea. It's a tool offered to NeuroMat researchers. Now it's open to any other researcher from the same department in Brazil as well. So far, we had, say, 5 Master of Science concluded, 2 ongoing. The 2 of them, one of them is here, the other one is English at the moment. 4 concluded PhDs, 1 ongoing, the PhD students here. 4 postdoctoral projects, 1 ongoing. I think Carmo is there as well, concluding. And 57 papers published, not only by my group, but by other groups as well. This kind of gives you an idea of the productivity of the center, of the Sing Lab. Here, let's see if I can pass it. Yeah. So why a sim lab in a neuromathematics center? Mathematicians would like to do everything on paper and pencil and paper, but well, the brain is a multi-scale system, as you can see here in this right-hand side figure, and computational models are particularly well suited for integrating the knowledge that you have, say, from channel scale, say molecular scale, into say large network scale. Because when you do a simulation, you have to, I mean, unless even if you use an approach like the one Victor presented to us, you need to have some detail, some understanding of the dynamics and the line dynamics in order to construct the large scale model. But if you do a model like the one which was presented by Marcus Disman early in this morning, you'd need at least knowledge from the single neuron dynamics and the connectivity and the large scale structure of the system. So this, if you put this in a simulation, you already have that in your model. You don't need to do any other assumption. You collect the data and use some hypothesis, and then you construct it. and then you simulate it, and then you see the effects, and then you can study the model by doing some interferences in some parameters of the model and see the behavior in a way very much like the experimentalist does with, say, real tissue. So that's one of the advantages of having a computational model. And in particular, in NeuroMat, we focus on large-scale models of cortical networks. So I'm going to present you a kind of overview of what we did over the years. So as Marcus Disman said this morning, we were the first. Well, we contributed largely to the establishment of reproducible modeling in computational neuroscience. We had the first full replication of the Potchens-Disman cortical microcircuit model that was done in Brian 2 by those guys here who were in my lab at the time. Those were, say, PhD and students, master students and PhD students. We did several re-implementations of the Potchens and Disman microcircuit model in different platforms like NetPine, which was also mentioned by Marcus this morning, and using different neuromodels, including the Galvez-Loscherbach model. And by doing this, we developed several international collaborations. There's one of them is mentioned here. This is the NetPine implementation. It involved collaboration with Bill Litton and Salvador Dura Bernal from State University of New York, and also the collaboration with people from ULI. And I was invited to participate in this community paper here, which was also mentioned by Marcus this morning. So this is one of the contributions we made. So this is something I mentioned this morning, and this is something that it shows the importance of having a computational model. We were able to, by using a stochastic version, this is the original Potchens and Disman model, a deterministic model. And this is the same, the model with the same anatomical connectivity of the Potchens and Disman model with Galvez-Loscherbach neurons, fitted by Vinicius in his master thesis. And you see, this is somehow not, you see the firing rate statistics and the synchrony and I mean the irregularity are more or less similar between the two models, but the synchrony measures are a little bit different. You see, for some reason it's not well, maybe it's the adapter there. The synchrony, there's some swaps there between the synchrony in the layer 2 and 3 excitatory. It's not related to this. And also the power spectrum between the two models, the deterministic one in magenta, the stochastic one in purple. You see there's a high frequency peak here in the deterministic model, which it disappears in the stochastic version. And this came out of the model naturally. So by introducing stochasticity, we lost this feature, which is, as Marcus mentioned, is not observed in real brains. So we don't have this high frequency picture. This peak. This is a feature of the deterministic model. That as we discussed this morning as well, was eventually eliminated from the deterministic model by introducing different types of neurons and changing the connectivity, the synaptic weights between inhibitory cells. So they found a way of eliminating that. But in our case, just by having the stochastic model, we were able to find this behavior very much as an experimentalist would do, doing experiments in real tissue. So this was one of the findings we had. Another one was when we replicated the full Pochantism model with ADAX neurons. ADAX neurons are like leak integrated and fire neurons. They are deterministic, but they are two-dimensional, and they are non-linear, and they have adaptation. And by doing this, by doing this, one is able to model different firing behaviors, like regular spiking, fast spiking, bursting. And we constructed, when we added to the connectivity model, to the Pochantism model, this neurons with different distributions between populations. Say in this case, the excitatory ones were regular spiking, the inhibitory ones were fast spiking. In this case, we introduced some bursting neurons in layer 5e. We changed the dynamics of the network. So the dynamics is sensitive to the type of neurons that you put in the model. And we know that in real brain, we have this, the neurons with different personalities, as we call. So this is another element that we were able to introduce and investigate in our simulations in the SimLab. Also based on international collaborations, in this case with Sasha Van Alvada from Jülich. Another type of study that we were able to do there in the SimLab was the study of phase transitions and criticality in networks of Galvez-Lochabar model. So the same type of study that Mauro presented to us this morning, initially using this simple network in terms of connectivity, this all-to-all connectivity with Galvez-Lochabar model. We were able, when I mentioned we, I mentioned those guys there, in particular Osami Kinouchi, were able to both find a way of analytically investigate the model and simulate it to find phase transitions and avalanches with different power laws there. And by later on, we studied networks with a more realistic topology, modular and hierarchical topologies, as you can see here. So neurons are grouped into modules. So within each module, the connectivity is stronger than between modules. And by doing this, we found we were able to observe that criticality is more efficiently maintained by networks with a hierarchical modular topology. So it's more robustly maintained by modularity than without modularity. So this shows that modularity has an importance, has a role in keeping the brain close to criticality, which is an important feature, as Mauro mentioned this morning. Also, everything was done in the SimLab, so that's why I'm showing this to you. Then we moved to construct by ourselves without using, say, the Pochantism model, which was constructed by people from Jülich, our own models for different brain regions. So one of them is the prefrontal cortex model. The prefrontal cortex is a very important area of the brain, so we decided to construct a detailed data-driven model using the ADAX neurons in our case. So not Galv's or Seba, but because of the fact that these neurons have they were able to simulate to replicate different firing behaviors with these neurons. So in this case, we had 10 different neuronal populations, so 10 different neuron types. 2 of them excitatory, 8 of them inhibitory. You had short-term synaptic plasticity in the model as well, and we were able to reproduce or to find transitions between different brain states. Asynchronous irregular, which is the most common type of brain state during wake behavior, and oscillatory states, which are very common during sleep. You see here, this is the asynchronous raster plot of the synchronous irregular state. This is a raster plot of the synchronous state. You can see more details in the Pedro's poster, which is here. Pedro is currently at EU. He's doing an internship there. So have a ongoing collaboration with people from ULI, and Pedro is our current student there. Oops. For some reason this is not working, so that's another large-scale cortical model we constructed, in this case by Rafael Gigante, who is here. This is part of his scientific initiation, finished last year, was a thalamocortical model, which had the parameters adjusted to replicate a feature found in patients with Parkinson's disease, which is a peak in the power spectrum in the beta. So the model was constructed to exhibit that peak, and then we simulated the effect of deep brain stimulation. Deep brain stimulation is usually applied to subcortical regions, but by antidromic connections between the basal ganglia and the cortex, some signals are propagated to the cortex. So we in our, this was just a scientific initiation, so just 1 year project. So we didn't include the basal ganglia in the model, so and included the direct stimulation by current input to the deep cortical layer in that case, and we showed by doing this we were able to eliminate the peak in the beta band. I don't know whether you can see here. Here you have for no patients without band, one, and so only after simulate deep brain stimulation. So you we found the reduction in the power in the beta band. Now Rafael, who is doing a master, is working on a primary motor cortex model, which will be say an improved model to be used in this cortical basal ganglia thalamic model that we plan to construct. So it's also a data-driven model based on ADAX neurons, as in the case of Pedro's model. 8 neuropopulations, again short-term synaptic plasticity, and again the model. Rafael was able to show that the model exhibits transitions between asynchronous, irregular, and oscillatory states. And naturally, interestingly, just by fitting the model to reproduce the firing behavior, the statistics of the firing behavior of the neuron, of the network in terms of layer-specific or population-specific firing rates, irregularity, and synchrony. Don't have time here to discuss the measures that we use, but the true measures, these are the standard measures that people use to classify synchrony and irregularity. But just by fixing this, the model naturally exhibited some high power in the beta band. So the model is kind of ready to be used in investigations of Parkinson's disease by simulations. So another type of model that we are studying there at our laboratory is an astrocyte model, which was introduced by Tiago Bezerra, who is here as well. So this is a very detailed, it's a compartmental model, conductance-based model of an astrocyte to be used in simulations where we have coupling between astrocytes and presynaptic and postsynaptic neurons in so-called tripartite synapses. This is a scheme of the model. See that it's very complicated, several channels, ion types. And since the model is a compartmental model, we were able to study the effect of the topology, the morphology of the single cell model to stimulation. And in that case, that it was a paper that came out in PLOS Computational Biology, we studied the combined effect of dopaminergic stimulation, which is a global stimulation of the entire cell, and glutamatergic stimulation, which occurs only at the site of at the trisynaptic synapse, the contact between an astrocytic process and a chemical synapse between two neurons. And we were able to show that topology of the morphology of the astrocyte, the way in which the processes are organized or bifurcate, influences the interaction between the two stimulations. So for further details, you can talk to Tiago or go to the paper. And then Tiago spent the past year in Finland, in Tampere, at Maria-Lina's Line lab, and there we constructed a neuroastrocyte network model to be used in large-scale simulations of neuronal astrocytic networks. In that case, we developed a simplified astrocyte model, a stochastic one, in which each, say, region of the astrocyte is modeled by an element which can be in 3 different states. Say like the SIR model, so it's stimulated, removed, and, or at the rest. So I'm just using words that you probably would understand better. And then by constructing networks of those astrocytes coupled to networks, we were able to show that astrocytes can modulate the neuronal activity. So here you have a raster plot of the network without the neuronal network without astrocytes, and then when astrocytes are coupled to the neuronal network and activated, you see here the green curve. This has an effect on the neuronal activity, so increases the neuronal activity just by the coupling between the trisynaptic coupling between the astrocytes and the neurons. So neurons, when they fire, they leak glutamate that affect the membrane of the astrocytes and lead to the entrance of calcium, and which are the activity measured in the astrocytes. So for the details, you can go to Tiago's poster here. So just a kind of summary of the main contributions that we had in the past 10 years. So we addressed reproducible cortical microcircuit models, which is a very important topic in computational neuroscience, because if we're not able to reproduce other people's work, what's the value of the work? So like in any type of science, you have to be able to reproduce the findings of one researcher using different platforms and different cell types. We studied stochastic and ADEX large-scale networks. We studied criticality and phase transitions, data-driven cortical models, neuroglial simulations, Parkinson's disease network modeling, and also training of students and international collaborations. For the next steps, which are the ongoing steps at the moment, so this is a kind of transition between NeuroMat and the INCT, the National Institute for Computational Neuroscience, which Mauro mentioned briefly this morning. We're using the we are investigating that interplay between astrocytes and neurons in a neuronal astrocytic network in simulated working memory tasks. So we also studying. Pedro is there in ULIS at the moment, so we are working with group from ULIS to construct a multi-columnar large-scale model of the prefrontal cortex, so a larger one, a larger network to study dynamic state transitions between, say, AI and synchronous oscillatory states. Rafael is currently working on extending his M1 model, the cortical model of primary motor cortex, to add the thalamus, as he did in his scientific initiation, and a model of the basal ganglia. You may not be able to see it there. And then we're not going to study DBS, but TMS, because we are in Ribeirão Preto and Buffer is as a TMS group there. So we want to be able to simulate the effect of transcranial magnetic stimulation on the model in a simulated Parkinsonian patient. And eventually couple so the astrocytic low-level neuronal models to the large-scale network models to have a multi-scale model, as shown here. That's the final goal. By having this multi-scale model, one would be able to bridge between phenomena at a, say, molecular level, single-channel level, to phenomena at a network level, and to try to understand the interaction between them in a mechanistic way. Yeah, that's my final slide. Thank you. PAPES, NeuroMat, USP, the school where I am, the department, and this is my group at the time, 10 years ago, my group now. So there was a reduction, but the department is more or less the same. It has been painted. Yeah. Okay, thank you very much. Questions?
I have two questions.
Yeah, of course. Yeah, there are people.
Can you make any?
yeah.
If I saw correctly, in the Marcus model, you have a shift in the small frequency peak when you put noise. Can you make any comment about that? If the
Low frequency, not high frequency.
yeah.
That's Well, I mean, yeah.
Yeah. Do you know why? Okay. And the other one is, can you make any comment about the self-organized criticality model? What have you modified in the GL model?
I had to go very briefly because I mean it's a
If
lot
it's-
of work.
There's time. If not,
Yeah, and the self-organized criticality.
Thank you for your talk, and thank you for also covering important things like criticality and very important terms. My questions will be mostly on the modeling and on the simulation. How do you take into consideration like you model, let's say you simulate the motor cortex for, how you take into consideration the functions? Because different people have different functions. They are not in the same spot as homunculus says. How do you take this into consideration? And how would you think that TMS would be a good input in this?
A good question. So this approach is different from the one kind of personalized approach. He's simplified at a single cell level by using mean fields, but they use functional magnetic resonance imaging data to construct, say, patient-specific brains, which allow you to, say, create digital twins of, say, single people. In our case, we're just using generic network models to try to capture some principles that would be useful for understanding, say, the mechanisms underlying Parkinson's disease in any human patient. So we are really far from getting as close as to, say, a single patient as Victor is with his type of modeling.
Okay, just I was thinking that the location and the connectivity of the location of the different functions, that they don't need to be only, simple movement, simple muscles. They can be coordinated movements, may play a very big role how to treat and understand the effect of Parkinson. That's why, and because of your relationship with TMS next door, I was thinking TMS is much more accurate than fMRI, okay? When we do direct cortical stimulation comparisons, fMRI has 20 millimeters errors, TMS has 5. So I would think that this kind of things can make them simulations much better.
That's right. Yeah, I agree with you.
Yeah.
The thing is, well, the construction of this type of model is something that really takes generations. We start, say, with a generic cortical model, just excitatory and inhibitory neurons connected in a random way. Now we have refined it to have a layer-specific cortical model with, say, populations as well, different populations in each layer, and then we try to reproduce what people observe in Parkinson's disease. So we twinkle with the parameters to have the high frequency in the beta band, the oscillations in beta band. So we assume that this somehow captures some mechanism, some underlying mechanism present in Parkinson's disease. And then we try to simulate, say, DBS or TMS, apply to that region to see the effects. The next step would probably use TMS to guide our construction of the model. This is something that still don't have. To do the verification. Then maybe you'll be able to have a digital twin of a given patient, because having the TMS data from that patient, one would be able to construct the network model. We're still far from that. We are, at the moment, we have this generic, say, microcircuit model in the style that people in Newlish are doing, and just to try to see whether we can reproduce the major findings. So some kind of stimulation of that region reduces the power in the beta band.
Just as an observer,
Yeah.
and coming from let's say, a good environment, infrastructure-wise, I see that you have everything. So it's you just need now to put the things together and to bring a lot of, let's say, people in to do experiments, because you have a lot of a lot of good things, a lot of resources, scientific resources to do these things.
We need these more people. You see, there were more people in the past than now. We need more and more students. That's it. We need to attract more good students. So we need hands to help us. So that's the main, one of the thresholds that we have now, the bottlenecks that we have now. Yeah, but you said, you're right. Yeah, the project would be like that. We need, we if we could get younger, we get younger by discussing this, but of course, our biology takes us to get old, so the younger people should also take, and having these international collaborations as well. Yeah. So we need to find collaborators to interact with this. Yeah.
Thank you. Thank you, Jorge, for this very, very nice presentation. Also, the whole session was very interesting, and our meeting is successful for sure. My question, now I'm putting the shows of Antonio Galvez, is yesterday when Florencia's the session, the mathematicians were speaking about those new models employing stochastic neurons, where you can observe this phenomena of this metastable phenomena that they are associated with plasticity and with learning, and so my question for you is, which would be the limitations on your point of view of employing these stochastic neurons for modeling? Because you employed this, you invested a lot in the understanding and looking at the circuits, but now when you look at short-term plasticity, for instance, you prefer using more simple models. So it's just
it's not simple models. Yeah, I mean, we are using these ADEX neurons because we want to have different firing behaviors. It's possible to have a stochastic model with different firing behaviors. We discussed that with Galvez and other students, collaborators at the time. So one can, because everything is in the phi of V function, the probability of firing. We can adjust that function so that it captures the firing behavior, say, of bursting neurons, rapid spiking neurons, fast spiking neurons, or regular spiking neurons. So it would be possible to have neurons with adaptation and also stochasticity present in that. So this is a task for the mathematicians to do. Yeah, the mathematicians are called, should be invited to join the effort and try to construct, say, improved stochastic models. We are working on that models because they are well established, and say life is short. So we're trying to approach, say, all the types of problems, say like constructing the large scale networks, getting the data to construct those networks, constructing the astrocytic models as well. So, but yeah, of course, with collaborations with other researchers who are more, say, skilled to construct the stochastic models, one would be able to improve. And yeah, but in terms of facilitation, in terms of, say, easiness of implementing, I mean, it's basically as easy, or even, say, easier to use the stochastic neural model. It's already at NEST. NEST is the main simulation tool developed by Jülich, and they have all the neural models that we use, and also the GL model. So it's something that we achieved during the interaction with people from Jülich. So what mathematicians could use the GL model, which is implemented in NEST, to do the simulations there.
Simulate this phenomenon.
To simulate large-scale networks, and add, say, extra neuronal models there, stochastic neuronal models that capture, say, bursting and other types of behavior. And then study short-term plasticity. Which, I mean, the same type of short-term plasticity that was introduced by Christophe and is the one we use here, because they are all based on Markran's idea that Zodix and Markran. So they have facilitation and depression. Facilitation based on the accumulation of calcium in the presynaptic neurons, and depression on the fact that as much as the neuron fires, it loses neurotransmitters to be used in future. So the mechanism, the underlying mechanism is the same that was implemented in both models.
But you expect, for instance, to see those metastable states in the model that you
the model exhibits that. Yeah. We actually studied it. I didn't show any slide on metastability, but we tried to do. But in that case, we used, say, generic random networks. We had random networks without any external stimulation, and then we just triggered the network with an external stimulation that given neuron, and then let the system evolve without any perturbation. And then we studied the statistics of decay back to the resting state. And then we found that there was an exponential distribution, very typical of metastable states. I've shown this to Galvez many times, and yeah. But the neuron, the single neuron was not a stochastic one, it was a deterministic one. Yeah. This is work that's still going on in my collaboration with other researchers in Germany and now in the States. A former student of mine, Rodrigo, is in the States now. They're pursuing this line of research.
Thank you very much.
Any other question? No? So we can close the session now, go for lunch, and then come back at 2 for the last session.
Som.
Good afternoon. It's a pleasure for me to work as chair this session. Our last session has as the main focus neurobiological and a clinical application. And our first speaker is our dear friend, Daniel Freiman, talking about the statistical comparison in brain networks. Thank you, Daniel.
Okay. Well, thanks, Antonio and Claudia, for the invitation. For me, it's a real pleasure to be here. I'm from most of the beginning of the NeuroMat project. I'm very grateful with Antonio Galvez that gave me the opportunity of meet the very nice friends and people here and great scientists. So I'm going to talk about some part of work we have done. And this year I collaborate with Claudia, with Florencia, with Gislan, with Edu Martins, with Michelle Miranda, with Antonio Galvez also. So for me it was a very nice project. And I'm going to tell a story about how we start with thinking about statistics of networks. And this story begins with a first paper about EEG functional networks that at that time Claudia and Edu works on this particular experiment where the people were watching a biological motion and a scramble motion. There was a very particular biological question about this, and they found differences using the ERP technique, that is like the common technique for analyzing this type of experiments. And at that time we asked where functional networks can give a complementary view of this type of difference. So what we did is to, with the same data, we analyze in a different way. Basically, we fix a sliding window. From this sliding window, we have an interaction criteria, and have like a correlation matrix. From the correlation matrix, with a threshold or another criteria. We have a network, 0 or 1, and then we study these networks that evolve in time. And we want to say something about if both conditions, biological or scrambled, were different. And the first thing is there's, as you can imagine, there's a lot of variability. There's a variability between trials and between subjects. And we found that if you study global properties of these networks, of these functional networks, we couldn't find any difference. This is scramble motion and biological motion. This is, for example, the average path length for networks of 30 links, 50 or 70 links. This is the clustering, the average clustering coefficient, and the other one is the average betweenness. But when you study local properties, like the degree of a particular nodes, we found that, for example, the electrode F7 has a smaller degree in the biological condition, or the similar the betweenness of the node. And for example, this electrode P3 have differences in the clustering coefficients, the number of neighbors that are connected. So the analogous of comparing these two conditions is something like this. From in time, you see differences, and this difference appears in particular electrodes and with different local properties. For example, this F7 has difference in degree and betweenness when you compare both conditions, and these electrodes, you see different things. Okay, so that were the result, and at that moment. And the obvious thing is, okay, how robust are the results? And fortunately, the experiment done was like that before the biological or scrambled condition that was randomly assigned. People didn't know there was a fixed cross. So we use the same methodology in this part to see if we observe spurious differences, and fortunately we didn't find any spurious. So we believe that this is robust. And we found an experiment where they applied TMS basically near to this electrode, the position of this electrode, and they found that they compare also biological motion and scramble, and basically they saw differences. So this confirmed that these results seems to be true. So at that time, the message was, okay, functional networks can be useful for analyzing this type of data. The information basically don't live in global average, they live in local properties. And the most important here is that this opens the door to develop more powerful techniques, in particular the ones that treat the network as a real input and not do like we done before, that is like, okay, see one-dimensional properties of the network. And with this in mind, we published 3 papers, one with Florencia and Andresa and Claudia, that I'm not going to talk about. I'm going to talk about the other ones, but basically opens one door there, that there was no, at that time, no methodology to compare networks. In particular, we always say that we are going to compare labeled networks, not networks that, you know, these nodes this node is associated with a particular region of the brain, not unlabeled. So basically, the bibliography was dominated by concentrate on individual network analysis. It's people that study networks know that, for example, community detection was something very important at that moment, and there were a lot of people working on new algorithms for that. But in that case, you have only one networks, and we know here in our context we have a lot of networks. So we concentrate on the analysis of a sample. So given a sample of networks, we are interested in have a unifying framework where we can define central variability measures, outlier detection, hypothesis testing, to do learning, supervised or unsupervised, and principal components. I'm going to talk a little about the three. So the key point in order to compare and to say things about networks is to have a metric. So if we have two networks, let's say G1 and G2, we use most simple distance, that is the distance or the Manhattan distance, that is just how many links are different. In this case, there is one link difference. And if we compare G3 with G1, it's the same, just this one. And now that we, okay, now choose a subject randomly and measure its brain activity, or its network. This is the random network. Yeah, the randomness comes from the way you choose the subject. And then if you know the law of this network, the probability of having exactly one of the possible graphs, you can compute any probability. In particular, for example, you can compute the probability of a connected graph. Okay. But once you have the law, the probability law of the graph, and the distance, you can define what is the expected distance from a particular graph to a random graph. Here, H is the graph, and G is the random graph. So this is a way to know how far you are from the, let's say, like an average graph. But the one way to define a central graph that will be useful is to say, okay, I want, if I have a set or a sample of networks, I would like to say this is representative of this sample. One way to do it is to central graph or the median graph, that is just the graph H that minimizes the expected distance from a random graph. Okay. So now if we have a sample of networks, we have this representative network. And if we have a representative network, we can compute variability. We should say, okay, compute the distance from each of the networks, or in the case of the population, compute the expected distance from a random graph. Okay. This is, if you know the law, you can compute this. But if you have a sample, you can estimate this just changing the expected value by the average. Okay. And these are consistent estimates. So now the point is, okay, there's a way to compute the central graph or the variability, but the point is that the network space is huge. For example, if you have 20 nodes, you have 10 to the 75 possible networks. And so how to find this graph H that minimize in that space? And the point is that it's very simple, because the the central graph satisfies that the adjacency matrix must verify that at least half of the case of a sample, half of the sample must have this link. So it's really easy to find this central graph. Just an example. This has nothing to do with networks. This is just dimension 1. You have not an exponential random variable and double exponential random variable. But you know, if you compute the expected value of this random variable, will be here at this 5. And you can do now the same with a graph. You can say, okay, just put here, you put the distance between a graph and a particular center. Okay. So in this is the probability law. This is S0, and we expect that if we compute the central graph, we obtain this S0, and if it's true, you obtain that. Okay. Now, if you have a distance, as I told you some minutes ago, it's very easy to know if you are an outlier or not, because an outlier will be very far from the other points. And an outlier can be an atypical brain network, or can be a non-healthy brain. So that says that if you compute the distance, you can say that. Obviously, this axis cannot be observed because it's one-dimensional, and the space of graph has no order. But you know, if you are very far, you will obtain values of this distance that are far. Okay. Any question? So, and now I'm going to talk about ANOVA for networks. And let's suppose we have 3 groups of subjects. One group, a control, I don't know, Alzheimer and DFT, dementia. And we want to know if each network is a brain, it's a person. And we want to know if the mean networks are the same, or if there is at least one difference between them. And we develop a particular statistic for this. That basically compare distance between groups and distance within groups. It's similar to what is done in ANOVA, in the one-dimensional ANOVA. But, so the first thing is here when you say equal mean network, what mean the mean network? The mean network is just the expected. This is the adjacency matrix in the component, the link A and J that connects node I with J, and we basically, this is the mean network, and we want to know if it's the same or not. And so let's suppose that there is one difference, at least one difference, and this is the sample. We have G1, G, and A from sample A, G1 to G and B to sample B, and sample C, okay? And let's suppose if they are different, we expect something like this, okay? In some space, in the correct space. So now, if we have a distance, we can, for example, compute what is the mean network here for this group, and compute the distance of this sample to its own center. This can be done for this own sample and for the other ones. And as you see, if the means are different, this distance will be much smaller than the other ones. And that is the idea that is behind. You can we do the same for a group, yeah, the same group, and then we compare this distance. Okay, let's suppose that now that H0 is true, so they have the same mean. They have the same mean. Basically, all points will be very near. The means of each group will be very near, and if we compute the mean of all together, we basically the same. So, we are going to compare all this distance for a unique central graph and compare that is this. That's why here says G and not GB, is GA. And we will compare these ones with when you do the same, but considering that you only measure the center from that. So in the case that the alternative hypothesis is true, we will observe that the one of the distance on the right will be much larger than on the left. And if it's true and the H0 is true, then you obtain basically the same. So the statistic is this one that compares, as you see, the distance computed from one sample to the distance of the joint sample. And we have the result that asymptotically is normal. You see, you have always average, so the central limit theorem will be valid. And now the point is, okay, let's suppose that we compare, let's put just two conditions, normal health control people and Alzheimer, and we compare, they are matched by age, we compare them, and our test says, okay, they are different. The value is, I don't know, -5, and you say, okay, the p-value is very small, they are different. Now the following question is, okay, where are the differences? The differences are in the nodes related with the frontal cortex, or okay. So once you detect, the next question is, okay, identify which are the links or the nodes that are the important ones. And so there are two types of problems that are very similar. One is to, let's say, this is all the brain. It has 5 nodes. And here, as you see, we are taking a subnetwork of nodes that may be the responsibles of this difference. And here we are selecting a subset of links that they are the responsibles of these differences. So you can try to solve any of that problems. But as we have this statistic, now we can say, okay, in this statistic there are information that is like the same in both population and information that is different. The information that is different is the one that is that give the evidence. So if you try to minimize modifying the network, for example, erase one node, okay, I erase this node, I compute the t-value, I erase this node, I compute the t-value and all that, there is one subset that is the best one in terms that the t-value is the worst. Let's say, okay, now you have the difference are the most notorious. And you can do the same in the case of the subset of links, okay? Select which links are the ones that contribute more to the differences. That's why here say, okay, the T is related with the links, and you find what is the set of links that minimize the T, or the set of nodes that minimize the T. This can be really a problem because the space is huge. It can be very difficult to find this. We have nowadays a solution for that. But the other one, it's not complicated. So, okay, that's the number of possible configurations. Okay, so now I will show an application of these results to some data, public data from Human Connectome project. There are 900 subjects, resting states of fMRI, and although the name is 900, there are 820 subjects, and for each of them, there is a lot of information. For example, there is more than one scan of the same subject, but you have individual socio-demographical, behavioral, and structural brain data. And each of these subjects has made some tests about cognition, emotion, personality. There's a lot of information. It's really nice data. And so what we did is here, all these subjects are control healthy people. So we invent categories, but we invent in this way. We construct. We don't invent. We construct in this way for each variable x that can be, I don't know, some socio-demographical. We separate the people between small x, middle x, or large x. Okay, so we have now 3 groups in this subjects. So the question is: are X and the brain network dependent? That is the same to say, okay, apply this ANOVA test. So this is part of the result. This is the variable X. So there is one variable X that is named, the name is inferior parietal thickness, right? And that is the thickness of the inferior parietal, of the right inferior parietal. And as you see, this is just to define networks, and you can define with numbers of links, with raw. But the important point is that we don't find any difference when we compare people that have different thickness, parietal thickness. But there are people that have sleep a little bit less, because one question was, how much have you slept yesterday or last week? And as you see, we found differences which are like maybe obvious, and we also find difference in temporal area. So now the interpretation, okay? So we can find what are the links that differ. It's not important here, but I think the interpretation is important here, because when we realize about this, of the results, when we study many of these X variables, let's say, okay, there is a lot of variability, and perhaps even if you let's say you are, you know, people that participate, okay, people that participate in this experiment, the experiment is just it says resting state. You are in a scanner, but okay, some let's suppose you are in a hospital, and okay, the scanner is there are some people using it. You have to wait like 30 minutes. Okay, do you want a coffee? Because he's coming. Sure, here's your coffee. Okay, so now, perhaps coffee is very important for the experiment. When we wrote that, a couple of months later appeared this paper that says caffeine causes a widespread increase in retinal brain damage. So the point is, there are a lot of things that influence fMRI activity. Obviously, the age, gender, education, and other things, as coffee. But one thing that we were very worried about is that we found that there was a set of 20 highly interdependent brain volumetric variables that were different, that gives different brain activity. So this put a problem because you want to compare, let's say, healthy people with Alzheimer. Okay, same age, perfect, same sex, okay. And now you go to the scanner, but this says, okay, no, the volumetric brain must be the same. And how do you know the volumetric brain? You have to put the people in the scanner. So you have first to scan, and then you have to select which of them you can compare because the volumetric variables are relevant in fMRI. So this, I think, is really a problem. And at that time, the important paper that says, okay, fMRI has many problems. The papers are not reproducible. We think that this goes in the same direction. So in summary, based on distance, we have robust network statistical comparison. This has a lot of applications. And in order to obtain highly reproducible results, you have to take care about confounding variables, that there are a lot. And I think it's a real problem. And recently, we are finishing another packages, so you can use it soon. Thanks a lot to NeuroMat team. And bye.
Thank you, Daniel. Some question?
Thank you for the talk. I'm not very familiar with graphs, so I don't know if my question is too naive, but in your adjacency matrix, the weights of the links are the distance themselves between the nodes, or no? Not necessarily.
Sorry, Daniel here.
How strong are the correlations? You can do it with the DTI, you can do also like how strong are the connections. But all what I have said is for for network that has the same weight. That is why here you see we always have like the same weight. You put a threshold and you change this threshold in order to, for example, this is a network of 20 links, and you see, okay, I don't see different. This is a network of 50 links. I don't see networks. So you can, they don't have weights, but we try to analyze as if they using like their weight.
Excuse me, could you
Okay. What I asked is whether the distance, what you call distance, is the sum of all links itself, and the distance, the actual measurement doesn't matter, right? Yeah.
And the real distance is.
So with your comment, what would be the impact of the volumetric differences between brains? That I don't get it.
But I'm trying to say something. But it's an excellent question to start. Osvaldo can start with that one. Now, okay. In order to do all this, you need to put data in a standard brain. And in this passage from standard to a standard brain, if you have, let's say, some parts of the brain that are greater, perhaps it is artificial there. That's the point. Perhaps the passage is artificial because you want to be like in the same scale, but I'm not sure, really. Or maybe that if you have like something that is thicker, then you have more activity there. If you have more activity there, then the links can be different. Why not? But
Well, my question is very simple. You mentioned all the details to take into account to have good data, including, you know, anxiety or coffee. But how about the differences in the equipment? Because to have such a large data set, you probably acquired the MRIs and different equipment. Do you know if they were checked to see if there was some consistency among them with, let's say, a phantom or a simulator?
Okay. I don't know the details, but I think that it's the same equipment because see here, they for the same subject they do 4 complete scans. This is not very typical. This is trying to say, okay, is how much variability we have even for the same scanner, but I'm not sure. But I think it's the same.
Good. Thank you, Daniel, for sharing your journey in NeuroMat with us. And I am very honored to introduce the next speaker, who are current NeuroMat leader, Osvaldo Paffa, talking about the legacy of CEPIT NeuroMat project in transcranial magnetic stimulation research.
So, good afternoon, everyone. I want to say it's a half pleasure to be here, right? I always say that we are experiencing a bittersweet sentiment because we are having a good meeting, but also probably be the last one of NeuroMat. So it's this kind of a ambivalent sentiment. But anyhow, I decided to talk about what were the achievements we're able to have in our group because we joined NeuroMat. And so it's concentrated in the magnetic stimulation, and I will show where we were when we started and where we are now. So you see that we got a lot of progress thanks to this partnership. And to start, I want to thank Galvis and Claudia. I think Claudia suggests Gal to invite me, and because of this relationship, we joined NeuroMat. And as I said at the beginning, I didn't know exactly how I could do it, but along the years, I think I found my place and will be able to contribute significantly to the project. Okay. So when we talk about the brain, there are many questions. How can we study the brain? And if you go to the library and look to Da Vinci's drawing about anatomy, you see that he drew very, very nice sketches about the whole body. But when it goes to the brain, that's what he saw. Just, this mass thing, greasy stuff there that nobody knows how it works. Of course, with time, many technologies appear, X-rays, magnetoencephalography, spectroscopy, and what else can help? I did include here the physiological measurements that were very important, right? And led us to learn a lot about the brain. So in a typical experiment, what we do is the following. We somehow stimulate the brain, and then there are all the detection area, let's say sensory organs, that will acquire the signal. And then there will be a processing area, and then we have the output. And this could be, as I said, electric signal, magnetic signals for the MEG. I can do proton relaxation. Daniel just talked about fMRI. I can measure oxygenation with near dot. That's a technique that Maria Elisa is working now. I can measure sugar consumption doing positron emission tomography. So all of these techniques is interesting. And there is one change in the paradigm that instead of stimuli the brain through the sensory organs, we can perhaps stimulate the brain directly. And this is what TMS allows us. We can talk directly to the brain because magnetic fields can pass the skull and reach the tissues there and induce currents, so we can stimulate direct areas in the brain. All right, that's what we do. And to do this very well, we need two things. We need precision and accuracy. We should hit the target and hit well. And that's the challenge we have now. Okay, I give you an example. This is very important in medicine. This is something I took from radiation oncology. People discover that, radiation can kill cancer cells. And then they start to treat tumors. So at the beginning, they have like a 2 kilovolt X-ray, and if you want to treat a prostate, you have to overlay the radiation here, so you get 40 grays here, and then technology improved, and then cobalt-6. So this is 1935, and then in the 50s, cobalt-6 came into place. You can see now that you can deposit a higher dose, and of course, when radiation crosses the tissue, you also produce effects here. And if you want to do a good treatment, you should concentrate the region in the tumor and spare the healthy tissue. So this is 50. In the 60s, the Linux appear, and then you can deposit a higher dose now in this configuration. And then now we have a 3D with tomography, and then we have now Linux with the intensity modulated radiotherapy, and you can see that now we can deposit a higher dose. And we are even changing the angle of incidence, so we can spare here the bladder and the rectal that are very sensitive to radiation. So this just to show how important precision is. And precision is a word that we use now every day. We have precision in mechanics that allows our cars to run longer, right? We have precision in agriculture that allows us to produce more food with less consumables and low cost. Okay, so how TMS works? TMS is a technique based on the changing of the magnetic field. So if this is Faraday's law, if I change the magnetic field very fast, I can produce electric fields, and the electric fields are linked to currents. So this is the way I produce currents within the brain, and I can stimulate the tissues. So this is the story. I mean, it started with some observations by Dunserval, thinking that he was seeing magnetic stimulation, but it was actually phosphines. And the TMS really appeared in 1985. So it's a technique that's now 40 years old, very new. And this is the guy, the team that invented it. This is Tony Barker, Ian Friesman, and Janolius, and the first equipment shown here. Okay, so what's the problem of having precision in TMS? It's shown here. Magnetic fields have this scene. Now, they start in the north and they want to go to the south. We don't have monopole. We don't have a magnetic monopole. They are always dipoles. And then you have the spotter, and this kind of smears the field. So we cannot focus in one region here. So if I want to, let's say, go deep, I increase the intensity here, I spread the signal or the stimulation to other areas. So that's the challenge. Okay? The things get more complicated when you look to the brain, because the brain is really convoluted, and then we have all this gyri and foci, and then you have to stimulate. And there is just one kind of electric field that will be able to stimulate the cells. It's this one that goes transversely, because it can produce a change in the potential of the membrane and then fire an actual potential. Okay? So to improve this situation, we have this developed that was done by Shogo Ueno. And instead of using just one circular coil here, he proposed to use 2 circular coils superposed here, so that we can increase the image current in this area, so we can have some focality. And this was done for quite some time. And my slides, I think, are kind of, okay, let's go in this direction. Okay, so this was a big step ahead in terms of improving the focality. Another very important development in the area was in the electronics, that people invented this H-bridge circuit that now can produce more pulses per second, so we can have a repetitive TMS. Another very important development was done by Risto Yumoniemi. That's also a partner of NeuroMat. He has been here several times. And he proposed to overlay several coils. And each coil has a different winding, so they can produce electric currents in different directions. And then if you combine those coils, you can orient the electric current in the brain, move sideways, rotate it, and do a lot of things. So all of these improvements in technology were important to achieve what we call precision and accuracy. Nowadays, there is the need of people are trying TMS in other structures. So this is the traditional TMS coil for stimulating the cortex, but some treatments need more deep stimulation. So people are producing those coils that look like a nest, so we can induce currents in the center of the brain or a more deeper structure to treat smoke cessation, obsessive compulsive disorder, major depressive disorder. So these are things people are trying now that has some success here and there. Okay, so the applications of TMS are shown here. We can using diagnostics for pre-surgical mapping, to measure motor evoked potential. This is something we do a lot. To study more basic brain functions, and in therapy, depression, and there are many clinical tests to apply in schizophrenia, obsessive compulsive disorder, Alzheimer's, and Parkinson's disease is one thing that we work here in NeuroMat with Maria Lisa's group. Okay. All right, so this is how TMS works. I have placed the coil in the cortex. I apply a pulse. Here you can see the pulse. The pulse generates actual potential that travels. And then I can measure here the motor evoked potential. And from there, I can measure the latency that tells about the speed. So if I have a demineralization of the nerves, the latency will be longer. If I lost some fibers, then the amplitude will decrease, so I can use this as a diagnosis. All right, so that's how we start. So these are the first measurements. You can see here Leonardo Rakauskas. Pantelis met him, and his looks very different now, right? But how we did those measurements? We did to do a mapping. So we place a swimming cap on the person. We drew a Cartesian grid there with several points, and then we manually place the coil in each point, measure the potential, apply a stimuli, and measure the motor evoked potential here, and made a map to see where is the hot spot, where is the area that activates the thumb. And then we start to do this, and soon we realized that was a very difficult task, because positioning here is very critical. You know, you place the coil, first you have to be at the right spot, and you have to keep the three angles, the pitch, the yaw, and the same orientation, and it's very difficult. So we said, well, we have to improve this, and then we found there were NeuroNavigators to do that, and they were very expensive. They were sold with the equipment, and once you buy it, if you want to change just one line, you have to pay a lot. It's closed, etc. So with the help of Andre and Victor, we are able to develop this software. That's free software, open access. You can get GitHub. Made a partnership with the Center of Information Technology, Renato Archer from Campinas. And so this was a big step ahead to improve our precision. And this is the things I just said. And this code was actually interesting. And even Google got interested in it and decided to give some support. So we had Victor and Renan, Thais, they went there to this Google Summer Code, they call, and they give some money to promote and bring people in the school to learn about it and contribute to it. All right, so now with this technique, we can do other things. We can apply pulse as before, measure now the electroencephalogram different points. I can measure the electromyography here, and we can produce maps like that. I say we, and we are trying. We are not there yet because this was, even though this was published in 1997, it's not an easy thing. And Professor Pantelis was here just to give us some lectures on how to do this more properly. But that's what we can do now with neuronavigation and these other things. So this is one study we did with undergraduate student, Gabriela. She's working in other area now, but we apply pulses in several areas of the brain using the neuronavigator. And then we collect the maps, and then we are able to produce areas of stimulation associated with the electric potential we measure here in the thumb. So as I said, the difficulties of TMS are positioning of the coil, slow process, operator dependent, and the accuracy and precision limitations, as I said before. Okay, then one time, Claudia was there. Rene was defending his master thesis. He just had finished the neuronavigator, details and put things there. And we kind of challenged him to go into this direction, right? And that's where we started, not too long ago. And then we decided to develop then a protocol of TMS that could use a robot positioning with single coils. And then we learned about the multilocus TMS. That's the name they give for the coil, the system that Risto developed that has several coils. So this was the idea of those projects. So I want to show you, this is a lot of complications as you're going to see. Same thing happened in radiotherapy there. You see, you have just an X-ray, and now you have Linux, MRI, and everything associated. So the more precise, the more precision you need, the more complex the technology is, and you need to be aware of this and be very careful. So this is the first robotic system we developed. Let's see if it works. Yeah. So you have the coil. These are markers that one camera is seeing. They are in the robotic. They are in the head of the subject. This is Renan during the pandemic. And you can see if he moves, the robot goes at the same position, keeps the position there, the same X, Y, and Z, and the same angles. So this was supposed to show the thumb moving. Okay, this is now the multi-channel system. You saw the stimulator there was just a box. This is not anymore a box. This is a cabinet. It has 5 coils here. So 1, 2, 3. It has 5 generators. So it's thinking that you are getting now 5 TMS stimulator piled in one top of the other. And you also have to pile on computer that should talk to each one of them and say, do this and that at that time. So that's what we have here. You can see it's much more complex. So we have transformer, electronics, and these are the capacitors that are charged, and then you discharge to produce the high intensity magnetic fields. And this is the system that was first built there in Finland, and showing the robotic also working there. Maybe I have to go there. I don't know the name of this guy. Do you know? Jonas, yeah. People told me he was the star subject. He had the best signal, and you see he moved it, and now the coil. Yeah, you have to. I didn't go into details about the robot, but it has a lots of safety features. Like if the subject moves, it has to goes away, goes in a curved path to approach. Because if I move the mark to the other side, the robot cannot cross here because it hits the head. So there's a lot of details here that I'm kind of skipping for the sake of time. So this is Thais now. She's here, so she can tell you what the experience is. And she's in our lab in Ribeirão, so we managed to, oops, oh, you already saw everything. The talks. Okay, let's see if it works. So through this collaboration we have with Aalto University, we're able to have this equipment in Ribeirão. And I think it's the 5th equipment we have in the world, right? If I'm not wrong. Yeah. So it's not moving. I don't know why. What do you mean? Yeah, I'm pressing. Yeah, I was supposed to work on the clip. Okay, but you know, it's the same thing. The robot will move, and but you can see now the markers are different. We are using those small spheres. Oh, okay, now it works. So this is the EEG instrument. We have, in this case, we have 2 robot arms, so we'll be able to do bilateral stimulation. So the neural navigator works like this. If you moves, then it goes to the same position. When everything gets green, then it applies the pulse. So with this robots now we call cobots, right? We can use TMS, multi-locus, and then using a second coil also. So we can do bilateral stimulation as shown here. And the idea is that the robot will take care of the slow dynamics. I mean, if the person moves the head a little bit, and the multi-locus can move from one point to the other very quick in times of milliseconds. I'll show you later. And this you can study other things that, okay, now we have so this is Renan. So they are the people that run the show. This is an experiment that we are using 2 robot arms, but different coils. This is the standard coil produced by MagVentor. And it's very important to wear slippers. We didn't have any. Yeah, just okay. With this multi-locus, as I said, we can do things like this. If you have just one coil like that energized, I can produce a electric field here. If I have another coil, it can go to here, to here. So if I superimpose all of them, I can produce a pulse here. And if I change the intensity, I can move the pulse to this position and from to this. So it depends on how I arrange the current. And I can move, as I said, from one spot to the other in times of the order of milliseconds. So this is very important to test networking and how an area talks to the other. There are many things that we even don't know yet, but could be accessed by this technology. And here, just to show you why it's important to do this technology development. So this is a paper from 1992 that was produced by Brazil Neto, and he was with Leonard Cohen at NIH at that time. And he discovered that by rotating the coil, you can change the intensity of the evoked response you have. So this was the first evidence that not only position, but orientation of the stimuli was very important, because as we saw, if we have the axons in this direction and I apply the current in this direction, nothing will happen. There will be no action potential. But if the current is perpendicular, then the membrane will depolarize and we have a potential. So at one point we didn't have a multi-locus, nothing. So we decided to use this multi-electrode sensor to put here in the thumb, and this was provided by Marco Antonio, the former student of Claudia, right? She was his PhD advisor. And then we are able to rotate the coil and make instead of controlling the pulse, we extended the area of observation of the muscle activity, able to make maps of superposition. And here is just the last work that Victor did by reproducing this experiment with the multi-locus TMS. And you can see here, they put in close to the central sulcus. This is 0 degree and 9 degree. And as you rotate, you can measure the amplitude of the electric potential evoked, I mean the map. And you can see that you have a much richer data here that you can do modeling better than what it was able to do in the form Brazil Neto and Cohen, but just 2 or 3 angles. So this, as you change the intensity, you can see the stimuli spreads around until area that you don't see almost any difference, right? Like that. So you have to be careful about as I said, the accuracy, the control, not only the position. So this is an experiment that's very preliminary. Thais did with the dual side TMS, and we could stimulate, let's say, M1 and M2. And as you change the pulse here, you can see the distribution you change. Here is another area to see connectivity, another experiment to see connectivity. One side, you be the right premotor cortex, the other M1, and you apply a conditioning stimuli and test stimuli, and you can also see difference. Anyhow, I think we are now in a situation where we have, I want to say the best, but it's one of the best facilities in Brazil to TMS. So many experiments, we are very well equipped. We need collaboration. We need people that could come with new ideas that could be solved by this technique. As I said, it's not easy. It's easy to talk, but when you go to practice, you have sometimes to spend 2 hours or 1 hour just to put the AG cap and get the right impedance and collect the data and so on. So everybody knows this. That work in the area knows this. And I think I was just talking to ChatGPT one day and asked a few questions. And so you see that I think we are in this direction of trying to have a neural navigation, to accelerate protocols, do personalized medicine. All of this is in our radar. So I will go. You can also talk to ChatGPT and get this. So I want to thank first the people. So without students, we don't do anything. So this is a picture they gave me when, I don't know, I think it was 40 years of the graduate program of medical physics, right? And so Lucas is working TMS, Thais, until recently, Joca, the team is more changes now. And some key collaborators are shown here. So Andrea was the first one that had the idea to start the neural navigation. He did the master in 2008, PhD 4 years later, and then Victor, 2014. And so Victor was recruited by Andrea. And then, so Victor finished in 2018, he's in Finland now. And then Victor recruited Renan. So, and then, this is the Renan. And then Lucas was finishing master's recently. Should be a C here, not an S. Here should be a C also. So Marcio is also here, Victor Moraes and Thais Marquette. And of course, I want to thank all the agents that support the project. So of course, first NeuroMat. As I said, NeuroMat was a key partner. Without it, it'd be much, much more difficult. You don't have we will experience this now because we all believe in NeuroMat. So you have to go to FAPEST with our own project every 2 years, and you see how the road will be difficult. University of São Paulo, of course, Alt University, partner, FAPESP, CNPq, CAPS, the Center of Technology and Information, Renato Archer, the graduate program in medical physics, the hospital, very important. I mean, we need MRIs, we go there, they do. We need a tractography, they do. They don't charge anything. And as I said before, we are now in this process of transformation. So CEPID is being gave birth to 3 ENCTs, right? So this is the new ENCT we have. We call PICS. Here is the name was Innovative Research in Science, in Health Science, from nanotechnology to artificial intelligence. We are very modest. Okay, with that I want to finish, and as the other speakers already said, I'd like to thank NeuroMat, thank the leadership of Antonio Galvez. I never imagined to be in this situation as I am before. And so thank you very much to all, and let's keep pushing. Thank you.
Thank you, Pafa. Questions?
Just curious. Do you know of any experiment that stimulate both hemispheres, and what type of? Yes, if you know of any experiment where people stimulate in both hemispheres at the same time, and what type of, I don't know, response, and what type of? Okay, for you then, right, if you want.
Quite a lot of effort on that. And But multilocus TMS is going to change this in the sense that the problem with this study is that you cannot study whatever you want. The coils do not fit. So now they are restricted for M1, left and right, and then you need to use also small coils. So the results are more variable than normally. So the multilocus is going to bridge this gap now.
Yeah, Bafa showed some pictures with the two coils. We did some preliminary experiments. As Pantelis said, it's very difficult to fit two coils in a person's head, but there are a lot of experiments that study the interhemispheric inhibition, so the inhibition that one hemisphere makes and the other one. And then depending on the time latency between these two pulses, usually you can change the response in the muscle. So you can highlight, for example, this inhibition and so on. I personally, or premotor areas or somatosensory areas. I don't know if Pantelis knows other.
For the moment, with the multilocus, it's M1 SMA simultaneously with different times. Also, I know that other colleagues with normal technology are doing parietal and prefrontal cortex. I do also speech mapping, and with double pulses like supramarginal gyrus and angular gyrus, and these things are preliminary. So there will be a lot of things coming out, but now we are on the, in the first. So you have to take advantage. Nothing has been done.
So there is one classical paradigm that if you stimulate just one side with a single pulse, then you just get one stimuli, one map. But then if you stimulate with two pulses, and if you change the interstimuli time, you can also have, one can be the conditioning pulse and the other the test pulse. Depending on the time between them, you can have a larger or a weaker response. So here you can see we did this not stimulating at the same site, but one stimuli here and the other contralateral. And then you see single pulse was this distribution. If the interstimuli was 4, was this distribution. So it's wider. So you have signals here that are stronger than the other. And here if it's 6 milliseconds, so it's also bigger. But if it's 40 milliseconds, then it's kind of inhibitory here. And then here is 160, I guess. So it has a distribution. So this was just with one subject. So we need to collect data from 10, 20 or more. Show that we are able to have this stimulation between the two hemispheres. So one side can talk to the other. As Thais said, head size matters here. Because this guy had a head big enough to put 2 coils, but many other people didn't have. But then what they found is that if you look to the connections, then you have in this case, the right ventral premotor cortex is connected to the other side. So then you can apply, not in the M1, but in the region here more frontal, that then you have space to put the coil. Then you can get a person with a smaller head. And when you do that, you see a pattern that also shows some changes when you change the stimuli. So we either have to produce smaller coils that are able to stimulate M1 and M2, or we can look to an accessory link to get there. Yeah, or with.
I have two quick questions. The first one, Do you think that the high variability from clinical trials could be associated with this physical, mechanical problem, the first one? And the second one, what do you think about the off-label application of this kind of stimulation in the clinical practice in Brazil? In Brazil, we are having a serious problem with this kind of off-label intervention. I would like to know your opinion.
Yeah, I think we are in a very critical moment in Brazil because equipments became affordable, and many people are buying TMS stimulators. And I think the practice now is, I don't know, many professionals can apply TMS. It's not restricted to medical doctors. So, and this is a problem because, yeah, physical therapists and I don't have nothing against physical therapists, but you have to know what you are doing, and people are now applying this to many things. So I went in May. I went to the physics meeting that was in Cuiabá, and I was there giving a talk, and then at the end, one guy that works in radiotherapy, he came to me and asked, "Well, why did this treatment and did not work? I have TOC, obsessive compulsive disorder, and then and why you did? Oh, my cousin did and worked for her, and for me not." And then I said, "Well, how they did? Well, they just placed there, and was $400 each session, and then 10 sessions for 1,000 reals, and then but did not work for me." And I said, "Oh, you're a medical physicist. You work in radiotherapy. Imagine that you are treating a tumor with X-ray source, and you don't have an X-ray image of your tumor, and you just by a palpation you put the X-ray there and treat. That's what you are doing. So it's the same precision. So I think it's mandatory to have navigation. I think we also should be discussing more about consensus on how to apply this, because if at the moment we have, let's say, a boom, a very good moment, people will do, but this can discredit the technique, and with time this will cost loss. You cost much more to recover the loss than if we do right from the beginning. But it's very difficult when you don't have such an organization that can control that. So thanks for the question.
Thank you very much. And now I would like to invite your next speaker. Oh, sorry. I am anxious for the next speaker. Thank you. We have 30 minutes of. Just a 10 minutes break. Okay, 10 minutes break. Here, we're talking about the neuroimaging guided TMS, TMS EEG, and the multifocus MS towards a fully automatic, individualized, and precise brain stimulation.
Thank you. Thank you very much, especially to Bafa, to Osvaldo and Antonio for having me here. It's a big honor and a big pleasure. First time for me in South America. And my talk will be about our efforts to scan the brain neurophysiologically, to create a neurophysiological connectome. And this requires, besides technology, a very high level of data quality. We want to understand the brain circuits. We want to predict brain states, pathologies. We cannot do it without AI and machine learning. We cannot do it with bad data. And this is the biggest challenge of AI and machine learning, the not understanding and knowing the data that people use. So I will start with my talk. It's a bit difficult to hold this and talk. Okay, so TMS is a great tool to map the brain, and we can map almost everything. I will show how, and I will show what this technology that you also have here, how it can be used and where it can drive us. So 41 years ago, TMS was introduced. It was a big invention. However, as you see, it was restricted to motor cortex. It could study the corticospinal excitability. Maybe it could study also the visual cortex. And it was an experimental tool. The very big breakthrough, though, came from Finland, from Risto Ilmoniemi. And actually, what you see a TMS that they were trying to make, and actually what they did was a mitigation plan because they failed making the multi-locus TMS, the multi-channel TMS then. But we managed now, so after so many years. So what Risto did, he introduced navigation to know where you stimulate. Immediately when you have navigation, you know where is your coil over the individual MRI. Once you have the individual MRI, you have personalized everything, almost. Okay, so that was the first thing. So I joined Biomag in 2004. They had already formed a company called Nextim, and it had already a neuronavigator. And we were using then, since then I'm using neuronavigation. I cannot do TMS without, there is no logic in that. So it allows us to place it reliably. It allows us to do test-retest design. It allows us to fuse different information. You can put data from other tools. So by having them individual MRI, you can actually study the motor functions. But now we don't talk about just studying. We talk about mapping. We don't talk about something general. We talk about the specific brain. So we can reproduce the homunculus for each individual non-invasively, already many years now. So this opened the road to use it as a pre-surgical evaluation tool. So already us in Biomag and in Germany, the colleagues from Charité, they showed that it's very precise tool, and it has an FDA license for TMS. Has FDA and CE mark for treatment of depression, motor and speech mapping, and treatment of OCD. So at the same time, I developed a method to study speech, to do the speech mapping. Now we don't evoke responses, we suppress the speech in order to show that this is a relevant speech related area. I'm not going to go more in detail into that. However, it's also an FDA tool. You need to name, you need to give the pulses at the correct spot, the correct time. The speech, it's a network. Okay, we talk about speech cortical network. Motor is also, the motor functions belong in a network also. So everything in the brain is a network. We cannot talk about one spot that is responsible for a function or for that when it's impaired, that it evokes a disorder. Neurological and psychiatric disorders are network disorders. Okay, this is a very important thing to understand. So yes, so we can have the coil, we can see where it is, we can know how much volts per meter the induced field can be in different parts of the brain, which is very important because there are different distances from skull to the cortex. We cannot use unified, and we have different set architecture. So many things. So yes, we can evoke an MEP, motor evoked potential. The time is not only related to health and pathology, as Bafa said, it's also about which muscle you stimulate. So the hands are from 17 to 25, the legs are later, it depends how long we are, everything. So this number cannot be averaged. People use 25 as an average. No, this is for you only. It's a number that, it's a personalized number, the latency of the map. So similarly, the latency of other things. So why TMS EEG? Without EEG, we can study areas like motor cortex with EMG, speech with behavior, visual with behavior. However, what about the rest of the brain? So many functions, so many things. Attention, memory, positive thoughts, negative thoughts, imageries. How can we study the excitability of the rest of the brain? So the only way is EEG, and that was what Risto did in 96. They created an amplifier that they are not anymore in out, but they were the only good ones then to record the EEG signal, because you get, with the TMS, you get also big artifact over the electrodes. So you stimulate, you get a cortical excitability, which we call it reactivity, the reaction of the brain to the TMS, and we care about that. We don't care about the multisensory things. But you get also the signal traveling. Okay, the signal travels. So this shows that there is some connectivity. We call it effective connectivity. That the one, if you stimulate here, it will travel somewhere. It depends on the wiring of the brain. It depends on the structural connectivity of the brain. Okay, in another area, if you stimulate, it will travel somewhere else. So Marcello Massimini, already then, with Giulio Tononi, they showed this unbelievable study. They stimulated here in pre-SMA so that they had good corticothalamic connections. And they showed that the signal, as Risto showed, is complicated and travels 280 milliseconds. When you are in full, when you are awake. But when you are in full sleep, deep sleep, non-REM sleep, the signal doesn't travel. It's not that complicated. It's bigger. It's higher excitability. The reactivity is bigger, but it has different frequency. So they proved that, and this now is used as biomarker to predict whether people will come out from coma. Okay. So this was called the breakdown of effective connectivity, that the sleep creates this. A pathology can create that. So one thing is this. Another thing that I was able to show during my PhD, because I had the tools, I didn't do anything very special actually, is that by stimulating two different cortical areas with 1 week difference each of them, M1 and DLPFC, we can get reproducible responses. So why these two areas? Because M1 is a good target for monitoring stroke and neurological disorders, and the LPFC is a good target to monitor mental psychiatric disorders. Okay, so and we first demonstrated that TMS can be a biomarker, actually, a monitoring tool. At the same time, the Italian team, Silvia Casarotto, showed that, okay, not only it is reproducible, but if you do small changes in the orientation, small changes in the location, or bigger changes, you can get completely different signal. So it's reproducible and sensitive, which means that it's an excellent mapping tool if you know how to use it. Okay, here you can see these two are different. The blue is here, the same volts per meter, but it has a very big, nice response. The other doesn't. You can see here that it looks like it has, but this is artifact. So if you do it correctly, then you see that with the same intensity, you evoke, you perturb the brain here, and if you go few centimeters forward, you don't do anything. You don't even touch the brain. You don't evoke the neurons, okay? So what the community is doing, they are finding the hot spot, they find the motor threshold, which is an intensity, the minimum intensity to evoke response, and they apply this everywhere. And that was the reason why, one of the reasons, not only location, that we have variability of results, okay? So what we see here is that we need for each, if we want to get the same response from all, we need to adjust the intensity. And then the same group showed something that I had seen already, but I didn't notice it. So you see here M1 has different shape than DLPFC. It's perturbed and then it goes back to the alpha rhythm, mu rhythm. So here is different. So what Mario Rosanova showed, which I consider maybe the best study ever with TMS-EEG, he showed this amazing thing. If you induce the same electric field, you get not only different reactivity, you see reactivity is the early response, but you get different shapes because they follow the natural frequency. So TMS-EEG can be a biomarker of brain circuits, of neuroanatomy, of cytoarchitecture. Okay. So, as I said, we need to, people use the motor threshold. The motor threshold is good for corticospinal studies. It's a corticospinal measure, so it should be restricted there. If I want to stimulate the visual cortex, I can use phosphins or I can use the EEG response. If I want to use the DLPFC, I need to use the EEG response. What's the problem? It has cranial nerves, so it's a bit difficult. That's why people do not do it, because they don't have the tools. So our job is to create the tools so people do their job correctly. Okay. So here this is a study, we gave 16 different intensities from non-perturbation to high perturbation. We were trying to find the threshold based on EEG, and we found also that if we want to do something like that, we need to use also the frequency, because this is also a way to recognize where you stimulate. So we are working towards thresholds based on EEG. And we can see here that in M1, the average can be, the threshold based on EEG can be close to 100 as 100% of motor threshold, okay? However, you see how different it is. In order to, we set a value called 6 microvolts because it's a good response if the background noise is about 1 to 2 microvolts, which is usually. So we saw that in order to get that, it's completely variable. So if I want to do a good study, even in M1, and make sure that I perturb the brain and I get a good response, I need to do it individually again. In pre-SMA, it's completely different. It's about over 120. And you see some have 160% of motor threshold in order to get 6 microvolts and the other 90 to get 6 microvolts. Why 6 microvolts? Because it's a value that if you give 150 pulses, you get it. If you give 20 pulses, you can get the same result as 150. So it's a good way to map the targets there. So anyways, we tried different techniques and different methods to find it. So this is the work of Ilka and Tida. So going back to Mario Rosanova's work, when I was trying to do pre-SMA studies for depression, we were trying to find a good target. I noticed something, that when we stimulate areas that they are very similar, how I noticed it? We put a coil, we give 20 pulses, and we see a real-time readout. Nobody's doing real-time readouts. Nobody's using this, but they do it for motor threshold. They do it for, why you don't do it for EEG? So with real-time readouts, we have a telescope and we observe what happens. So I didn't read to come up with this idea. I saw it. So I saw that they are different. Like less than, I'm not very intellectual, maybe, like a small move of the coil. So then we saw this and we published a proof of concept study, and then we studied it further. So we used different parts of pre-SMA and SMA. So why pre-SMA, SMA? Because they have very good corticothalamic connections and because it's considered the same area. Cytoarchitecturally it's the same, but we see differences. How are they explained? What is the thing here? And which is the best target? So we saw differences in 25 subjects, 22. And then we saw that the early responses across these targets, the blue is the most anterior, the red is the most posterior, they are significantly different. Almost all of them, the one with the others. Not the later responses after 50 or after 100 milliseconds, because these are more the traveling wave. And not only this, we saw that the same targets, okay, they had differences in alpha rhythm and in B2, but not in other frequencies. But we saw this, that they are, when they are compared, they are compared to each other according to their structural connectivity to thalamus, they are very different. And not only thalamus as one structure, we saw differences of each of them with different nuclei, thalamic nuclei. And the thalamic nuclei play roles. They regulate different functions. So maybe we can use this as a new way of targeting. Okay. So, and we saw also that the same targets have different connectivity to different cortices. As also Bafa was saying, that we may want to stimulate how structural connectivity in neuroimaging is a great tool. We need to use it. It will drive us. It will show us some things. Okay. The work from Francesco, Ali, and Fiammetta. And now, let's go to the biomarkers of depression. We moved the coil within the SMA complex, and we had differences. This is a big area. If we want to do the same in DLPFC, and why we want to do this? Because if I place the coil in a standard way in BMF3 that psychiatrists use as treatment, I will have 8 out of 10 times huge artifact. So I don't want to record the artifact because when I will need to remove it, I remove brain signal. If I remove brain signal, I remove also the power of TMS-EEG to be used as biomarker. I remove the predictive power. Okay, so what we thought that let's map, let's go away the targets, but within the LPFC, and let's find a good spot so we have better predictive power. So we used the parcellations from human connectome study. Then we used fMRI that the anticorrelation with area 25, which is the main hub for depression. And then we used also this so-called real-time tractography. And then we start mapping. So we gave 20 pulses here, 20 here, 20 here, 20 here to get the average of TMS-EEG response to see how it is. And we compared it with the standard target. The standard target, because it's used with 120% of motor threshold, we used a variable intensities, just to make sure that we get a big response, to make sure that I perturb the brain. This is what I care. Before I record, I want to know that I have managed. As nobody starts any study if they don't find the motor threshold. A similar thing. So here you see that by adjusting, this is the original response, if you are lucky. And then by adjusting it with the same even intensity, you may make it much more reactive and better. So you see the later responses, do you see this? They are similar because it's the same area more or less. But the early response, the reactivity is different. The same thing as with the pre-SMA, SMA. The later responses were not significantly different because the areas are connected in a general way, most probably. So anyways, we found out that we can use this and we can correlate it with symptom scores, with behavioral scores. We are now trying to publish. It's a bit difficult to convince the reviewers about these real-time things that we compare the different peak-to-peaks. And my target is that what I want to do is that we put the coil, we give 20 pulses, we find a good spot, we give 20 pulses, and we say, okay, this is a patient, this is not. For this, I need a lot of machine learning, a lot of people, but we have an idea how it works, a lot of expertise. And we need to go away from that thing. So we can also use the TMS-EEG to characterize tumors. So this is TMS-EEG inside the tumor before resection. After, this is perilesion before resection. And this is the healthy tissue before resection on the other side, contralaterally. And this is after resection, after 2-3 months, everything recovers. And even the other side becomes even more excitable. So we would like to characterize the tumors to understand where they stop also. Where is the lesion, where is the perilesion, and then see also, decide what kind of rehabilitation or who needs what. Okay, so it's a long-term project, this. So now, we are in this situation, we give a pulse, we move it manually, we can get the topo plots, we can get the evoked responses, but it's manual, too much, limited accuracy. And when we talk about mapping, it's also limited reproducibility. We cannot get exactly the same spots. Maybe with a robot, yes, you can. But it's very slow with a robot. So it's slow also manually. The biggest problem with TMS is this. We are the problem. Variability is a good thing in the brain. It allows learning. It allows developing. What is not good is the operator-induced variability. So this is what we are trying with all this effort to stop and to, not to stop, to change, to prove and to change. And of course, it's blind to the brain state. So multilocus allows high resolution in location and orientation, and you can give pulses with many milliseconds, within milliseconds. If you have something like that, you can stimulate it. Because as Bafa showed, the culture, channels there. It's not multi-channel, it's multi-locus. So what we want to perturb, we want to measure, we want to use EEG feedback and adjust. Okay, 5 minutes, good. So this was shown by Bafa. This video was shown by Bafa. This was shown by Bafa, but I will show it a bit differently. So when we put a current like this, we discharge a current like this in this coil, we get induced field with this orientation. Different coils, different electric field distribution in the brain. You see all these are different. Now, if we start combining, we can electronically shift, okay? We shift it even more. We engage also this. We can rotate. So we can actually scan neurophysiologically. And we can do motor mapping for pre-surgical evaluation faster, more reliably. We can do these things. We are close to finish the studies and prove these things. So it gives us very big advantages. So it allows to find the hot spot automatically. And what is this advantage is that we in Finland and you here, we can do clinical trials in exactly the same way because we will have the same algorithm. This is a very huge thing. Like this is like, of course, stimulating 2 and 3 spots and going deeper is a very big thing too, but for clinical trials that you were mentioning that the results are not, that's why the results are not. But now if we use the algorithm to find them, the hot spot and the intensity and everything, at least we unify the error. Okay, at least. So by doing the same mistake, it's... So we have real-time, we have realistic models of the head. Victor has done, and Renan have done amazing work with us also. So, as we said, there is the state-dependent TMS. So you can stimulate, the EEG can trigger the TMS. So if you want to stimulate in new rhythm in the positive or negative spot, you can do it. The EEG will trigger it. And then you can do it also with multilocus, like you want frontally to be this rhythm and parietally another rhythm. So we can do, in theory, a lot of things. I worked with spikes. So we did a study predicting spikes and stimulating during the spikes, because this is also a brain state. Okay, I'm not going to be in this, and I will show this video, which is the pre-SMA mapping. You see, it doesn't move. The robot just holds it and makes sure that it's always in the same spot. Then we shift electronically the pulse. And what we can do, we can optimize for this area, let's say, which of these locations gives the biggest response. But we can do also the other thing. Which of these locations optimize the response remotely? So this is the work of Ida. And here are some members. We are a big team, Risto, of course. Victor, of course, Ida, of course, Tuomas Mutanen, Matilda, Anna Del Soto, and me, mostly looking. Not really. I'm very much hands-on. I don't consider science without doing experiments. Okay, it's not, they're not on the computer. So also on the computer. So, and last, I would like to show how brain state kind of condition, the stimulation can be used for therapies. So we use the motor execution here, but it can be motor imagery instead. So he moves the left hand and the robot moves to the right side. So what does it mean? That we can trigger the TMS based on motor or mental imagery. So we have the platform that was, we are thankful to Renan about it and to Matilda. So you can actually, stimulate your brain with your brain. And this is a very big thing because we can do therapies also in places that are very far and there are no experts. We can install systems. So that's all, I think. Yes.
Thank you. Questions?
Thank you, Pantelis. I still don't get how do you do the trade-off between the functional targets using the fMRI with SGACC, with the LPFC, and with the other targets that you mentioned to optimize the TEP itself. Like, because what I mean is that you kind of stimulate a broader area, I believe, than the exact voxels that has the anti-correlation with SGACC.
Me.
So we do that. The question is good. But anyways, this study is about many targets, okay? DLPFC, DMPFC, pre-SMA. So when I do TMS-EEG for biomarker, what is the most important thing is this, the signal. How I can predict anything with this? This is, look what it is. How can I predict? So if I don't have a signal, I cannot do this thing. So my trade-off is that I go a bit further, but still architecturally relevant. You know, in M1, if you move the coil from the motor knob a bit to the right, you are still in M1. But you have a bit different, maybe, function. It's another thing. So something similar might be there, but because it's a bit broader and because it's an overlap, we may be relevant and we may predict. So I have another slide, which I don't show, what I believe is the best way of doing this, TMS fMRI. Because you can see everything. But now here, what we did, we tried to make this as big as possible. Isn't it logical with this? I can have bigger predictive power than with this. So this is the assumption. And nobody knows that this spot is accurate and correct. Okay, it's a statistical correlation. It's not proved, and we don't know, like we don't have direct connections. We don't know if they exist. Like we don't know if there is structural connectivity between these two. So even pre-SMA is a very good target for, I was talking with Helen Mayberg, who is the one that found the area 25, and she explained to me that because of corticothalamic connections, you can get it also from there. So this is for biomarker, it's not for treatment. And if I wanted to find a good spot, the best for treatment and for, I would do it with TMS fMRI, with multi-locus TMS fMRI, because then I can map. Because now you cannot map inside the fMRI, you need to bring inside and out multiple times. We can discuss it.
Thank you for the presentation. I would like to know how much do we know about the relationship between the motor evoked potential amplitude and the early TEPs, actually the early components of the...
We know. That's why also it correlated a bit the motor thresholds. There is the study of Hanamaki and Risto Elmoniemi, 2000, I don't remember, that it found big correlation between the 1530 response and the MEP. So also, like, common sense. We don't need even to question. I will show you this. Because I don't do talks, I do also a bit of teaching, preaching. Opa. So, 23 to go down and then another 23 to come back. And a lot of ringing in between. So, if this is 18, most probably the N35, the 35 response in the TEP, has also contribution from that. For this one, 23, it's the 46. The contribution is the 46. So there is a very good relationship between those. But the thing is, how you use this to stimulate M1, so you consider that this is your effect, TMS effect to the brain, I agree with you, and how you don't think the same about the EEG. Why I need to prove it? I don't need to prove it. I don't need to, actually, the truth is that it's even earlier. It's 2 milliseconds, 3, but it's covered. So the closest to that is the 10 milliseconds that we can see. Okay, the problem is that people learn to follow trends because there were no tools. And now the simple thing, I don't do anything, guys. I don't do anything. I do very simple stuff. And I need to go to the question of yeah. So I try to get a bigger response. Wouldn't you do the same for motor threshold? The same you will do. Why you don't ask this and you ask this? I don't understand. I'm trying to evoke, okay? It's to evoke the Socrates used to do this.
I have a provocative question, okay? Do you think that this kind of precision approach will be scalable in the future for clinical practice?
And like, if I go to do these things, we did it with patients, but if I'm gonna do it for every day, my students hate me about the study. Okay, but I did also myself a lot of experiments to help them. So yes, no, it's not logistically feasible. So, but we have the multi-locus TMS, and we have the robots, and we have the algorithms. So we will shift it there. So first we try to demonstrate that there is a reason in doing it. Okay? Yeah, but no, it's not feasible for the moment. We will do it through the new technologies.
Maybe a Brazilian robotic brace, low cost, user friendly.
And to have very good algorithms, but we need a lot of work. Like, these things will be ready to some extent in 10, 20 years. Like,
could be challenging for the next 10 years. Thank you very much. And now, our next speak, Daniel. We are ready? Yes. Yes. Ah, okay. I look. I am waiting, Arthur. Okay, we are right? Okay. I'd like to introduce the next speaker, Daniel Takahashi, talking about inferring information from in-high-dimensional system from incomplete and asynchronous observation. Welcome, Daniel.
Hi everyone, can you hear me? Can you hear?
Yes.
Okay, good.
Okay.
Sorry, it's really, yeah, it's sometimes
Welcome.
it's difficult to give a talk online, you know, you don't get the feedback. So hi everyone, good afternoon. First I wanted to thank Hockey, Claudia and Bato for inviting me and giving me this opportunity to talk a little bit about our recent work. This is actually my first time talking about this work, so hopefully it goes well. So today we'll talk about a method that we developed in the framework of this NeuroMat. Right? And this work was jointly done with Isi Zhang. She's from Tsinghua University in China, in Beijing. And the motivation is the following. Nowadays, I think it's a commonplace that people are trying to record in a very large scale way. Right? So for example, nowadays we have these neuropixels that record from thousands of neurons simultaneously. We have also calcium imaging that can record 1,000 neurons at once. And also now we have ECOGs that can record from wide brain areas in humans. And when you look at the literature, more and more what's going on is that several groups are joining efforts to record using this recording device that already alone, it's a very high dimensional recording device, but now they're combining and try to get something that is more holistic view of what's going on in the brain. So for example, in the neuropixels, we know we have now several groups that are joining efforts. For example, there's the famous IBL, the International Brain Laboratory, that a lot of different laboratories are recording several different brain areas in the rats or the mice, so that and they're sharing the data set so that we can actually use this data set. Also, with the calcium imaging, a lot of groups are imaging in different brain areas in the rats or in the mice, and also there are a lot of data set that is shared nowadays. And the ECOG is the same, or ECOG or the intracranial EEG is also the same. So now there are several groups doing similar kind of recording and they're sharing the data set. But there is now a fundamental problem, which is the following. Now we have a lot of data sets with a very high coverage, but all these data sets were recorded in different laboratories, meaning that they were recorded not simultaneously. Right? And then the problem for us is that if the data set was not recorded simultaneously, there are several things that probably we cannot, or it seems to be difficult for us to infer. And one of them is the information flow between these brain areas. Right? And what's the problem when it's not recorded simultaneously? And this is the most the simplest of those problems. It's actually the spurious causality problem. Let's say that you have X1 and then X2, and you just record these two, but you don't record the rest. And then when you try to infer the information flow between these two areas, what's happening is that you think that there is a connection between X1 and X2, but in reality, maybe there was another area, X3, that actually was sending information to the both. Right? And in this case, what's really interesting that is not that you didn't record X3, right? It's just the problem that you didn't record simultaneously. So even if you record the X3, maybe you recorded even X3 and X1, X3, X2, and X1 and X2 in different instances, let's say in different sessions or different laboratories, but you didn't record simultaneously. And if you didn't record simultaneously, in principle, you could miss and make wrong inference. Is that clear for everyone? I cannot see. Okay. Okay. So now the question is the following: can we actually overcome this problem? Okay. And what we will show is that in most cases, at least in the case that people consider in the literature, pretty much we can actually overcome this problem. And that's the idea of the method that I will introduce here. Okay. So here, just to see, so there are several methods. So for example, there is a gradual causality method. It will give some information flow ideas, but it cannot operate without simultaneous recording. It's in principle, it cannot handle unobserved region pairs, and then also it's a parametric model. So for example, if you want to analyze spiking data sets, right? It could be difficult to use the naive Granger causal. The dynamical causal modeling is also quite similar, right? There are other methods that is designed exactly to when, in the case that you didn't record simultaneous, but those are mostly correlational methods. Okay? So the idea is that we are proposing this method that we are calling SPIDER, that essentially can satisfy all these four. So essentially, it will work even when we cannot record everything simultaneously. It will handle unobserved region pairs. It will yield direct information flow, and probably most importantly, or very important, it's also a non-parametric method. Okay. And again, so here's the problem. Let's say that you have several recordings from several brain areas, but they are not aligned. Right? Obviously, if you have like something that make the clock to reset, let's say, you have the same stimuli to everyone, that will be enough. But that's not the case. In the most of cases, let's say that you are trying to analyze the resting state brain, you don't have any time locking stimuli. So the question is what you can do. And what's really interesting that in the time domain, it seems to be difficult to solve this problem. But actually, we will show that in the frequency domain, that can be solved. And the idea is quite simple. Right? The idea is to use a fundamental property of the stationary process that if you consider the frequency domain representation, right, once you have the entire frequency domain interaction, pairwise interaction, it's enough for you to define uniquely the random process. So how it will work? So let's say that you have a synchronous block of recording. And then what you will do that you calculate the power spectrum pairwise, okay, the cross spectrum pairwise. And although you didn't see simultaneous everything, in the total, maybe you saw everything. Then what you will do that you will complete the entire matrix by stitching the different power spectrum. What does it mean? So let's say that one laboratory recorded from area 1 and 2, here and here. Can you see my arrow? Yeah, you can see. 1 and 2, right? And you form this block. And another laboratory observed these 2 blocks, then you can see it here. And then let's say that another laboratory recorded this area, right? So this interaction. So now in total, you have all the pairwise interaction. And the idea that once you have all the pairwise interaction, you can actually, there is a procedure for you to recover the causality from this power spectrum. Okay. So here is exactly what I just said. So let's say that you have one session or one laboratory to record here, another laboratory to record here. And the interesting is that here they both recorded the same thing. Right? So they have something in common. Okay? And now the idea that just the fact that you have something in common, you can recover what's missing and you can reconstruct all the interactions. And this technique works even in a very sparse, even when the recording was done in a very sparse situation. Let's say that actually you have several laboratories, but it was not enough, for example, to cover all the all the possible pairwise interaction. So there are a lot of missing interactions. Even in this case, actually, we can show that we can actually recover the interaction that are missing. Okay. And then we have a term that says that essentially the recovery rate depends on the rank of the matrix. So it's really difficult sometimes. So if you have any question, you can also ask in the middle. It's a little bit difficult without the feedback. I'm not sure. Is it clear so far? Yeah. And once we have, actually, once we reconstruct the power spectrum matrix, what we can do is to, once you have this reconstructed power spectrum, the problem is that the technical problem that this power spectrum usually is not positive definite, okay? And if it's not positive definite, we cannot apply actually the algorithm to recover the causality. So that's the gist. So then you have to somehow fix the fact that it's not positive definite. And what you do is actually you use a G-lasso, a graphical lasso regularization method to make it positive definite. And once you have that, now you can actually recover the causality interaction from the this power spectrum matrix using two steps. Right? So this Wilson algorithm, and then use the coefficient that you got from the Wilson algorithm to calculate the partial direct coherence, which measure actually the information flow in the frequency domain. Okay, and what's really interesting is that this part is entirely non-parametric. Right? It means that we can use for EEG dataset, LFP dataset, spikes, and even when it's a mixture of spikes and LFPs. Well, another thing that sometimes we note that it becomes a problem is that a lot of times, for example, when you have this neuropixel, you insert the electrode, and several electrodes are in the same area, brain area. Okay, and they have a lot of very high correlation between them. And usually what you want is something, it's the information flow, not between exactly one neuron to the other neuron, but between one brain area to another brain area, right? And in this case, so the question is that what does it mean to have information flow moving between one brain area and another brain area? What you have to do is to somehow calculate something similar to the canonical correlation. So, and we developed a method to calculate the canonical correlation between brain areas in the frequency domain. Okay, so essentially what I mean is that now with this technique we can say what's the amount of information from one brain area to the other brain area, even if you have several recordings from the same brain areas. Okay, so you can uniquely define what is the information from one brain area to another brain. And there is these two situations that are important to emphasize. One situation where actually, when you consider all the possible data sets, in reality, you recover all the pairwise interaction. In this case, you can do this by design. Now, let's say that actually you are recording from different animals, but you are recording in small brain areas in each animal. But if you consider all the animals together, you have actually all the possible interaction. Or sometimes you cannot do that, and then you miss a lot of brain areas. Right? That maybe sometimes is more realistic, but obviously in general, in this case, you can get a better recovery than this. But our methods can deal with both cases. So this is a first example. So we model Izekevich neural network with 2 neurons. And we assume that we only look at 2 neurons at a time. So we simulate a situation that we record only N1 and N2, only N1 and N3, N2 and N3. Okay? And then here is the graph of the synaptic strength in the simulated model. And here is the information flow that will recover by only looking at the pairwise interaction. And you see that what we recover is a correlation of almost like a 1. Right? So you get a really good recovery, although at each time we only observe pairwise. Okay? We never observe all 3 simultaneously. We also simulated in a much bigger network, and then we tested how much of the network can still be missed and we can still recover. And we find out that more or less it's 30% is the threshold. So if we miss around 30%, our method still can recover the entire network, although we never observe the 30% of the interaction. Okay. We also apply now in the real data set. So here is it's a calcium imaging where we have 49 neurons. And in this case, obviously, this 49 neurons was observed simultaneously. And what we did was actually to, on purpose, erase 37% of the interaction and see how well our method, the spider, would recover the missing interaction. And what we found out, that even if we erase 37% of the interaction that's that exists, we still get a correlation of 0.86 using the spider. Okay, the correlation was 0.86 of the interaction strength, compared to the original, when compared to the case that we observe everything. And finally, we use to the International Brain laboratory data set. So in this case, we had 12 separate laboratories, 39 adult mice, where they recorded pretty much everywhere using neuropixels, thousands and thousands of neurons, right? But they were not recorded all of them simultaneously. That's the important point. And what we did actually was to use this method, right, and try to recover the interaction between these brain areas using our method, the spider. And what we found out first is that we compare with the anatomical connectivity from the Allen Brain Connectivity Atlas, right? So the physical connectivity. And what we find out is that we can find arrows in the area under the curve of around 0.9 for our method compared to the physical connectivity. And again, what's really interesting that none of the laboratory recorded a huge brain area. So we had to stitch all the 12 data sets from 12 different laboratories to get this connectivity. And now we can ask interesting questions. So let's say you are interested to know if actually the brain is somehow hierarchical or it's mostly feedback loops, right? Because now you have essentially the network of pretty much the entire brain using just the data sets from International Brain Laboratory. Yeah? And we did this, and what we found out, so here we measure the non-hierarchiness at 0, and essentially when it's low, it means that actually, so it's a little bit confused, but when it's low, it means that it has a lot of hierarchy, okay? And when it's high, it means that it's not hierarchical, it's mostly feedback. And what you see is that in this brain, when these labs were not doing anything, they were just like doing random things, right? They were resting in some sense. And what you see is that in the theta band, right, around 4 to 8 hertz, you see a very strong hierarchical structure, although in all the rest of the frequency, you don't see this hierarchical structure, okay? And we can see actually that the area that is sending the most information, actually it's the hippocampus. So the hippocampus seems to be in the top of the hierarchy, and the last parts of the hierarchy, the one that is receiving the most information, at least in this data set, seems to be actually the PAD. No? So it's quite interesting. And now we also apply exactly the same method in humans. And we got exactly the same result. So we found out that even in humans, in the theta band, the hierarchical signal is much stronger than in the other figures. Pretty much in other figures, it's mostly feedback loops, but in theta band, it seems that the information has a very clear hierarchical structure. Okay? In this case, we didn't have deep neurostructures, so we have mostly actually cortical structure, but what we see is that limbic structures and paralimbic structure were actually the source, right? And the primary sensory areas were actually the one that received the signal. So they were modulated by this limbic structure in our case. Okay. So now, with this method, we were able to essentially use all the data sets from all these different laboratories, combine them to have a brain-wide information flow map that doesn't have that previously was not possible because you were not recording synchronous. Right? So now we overcome this problem both in rodents and also in humans. And we found actually a reproducible pattern where actually in the theta band we see a very clear hierarchical structure of information flow. and I didn't show because mostly because I don't have time, but most of the actually the results is proven mathematically, and we have now an article in the archive where everything is proven there. And with this, thank you so much. I wanted to thank really NeuroMat for all the fundings, collaboration opportunities, and all the friendships. And Antonio Gauss, my friend, you have raised this. Thank you so much.
Thank you, Daniel. Questions? A minute. Ladies first.
Thank you, Daniel. I may have missed it. Could you please revisit the idea of hierarchy in your model? How
Yes.
you define this? Thank you.
Yes, sorry that maybe I went too fast. So the idea of hierarchy is that there are feed-forward connections. So because in our method we can find feedbacks also, but when you look at the two structures, if there is one side that is much stronger connectivity, well, it means that there is a feed-forward network. So when you say hierarchy, it means that most of the pairwise always there was a clear directionality. So essentially, you can follow one by one, and you can see where in the hierarchy you see. So when it's feedback, you cannot do this because now you have loops, and there is a way to quantify that. It's called the incoherence index, or here I'm calling the non-hierarchicalness. It's a little bit mouthful, yeah. Mouth, yeah. So we can measure this, and then once we can measure this for each frequency, because now we have a connectivity for each frequency, and we find that in theta band, actually, most of the connection was actually, it's one-sided. That's what I did. Yeah.
Thank you, Daniel. Daniel, now here we will have a coffee break, and I hope that you have a good coffee here.
Yeah, thank you. Okay.
Next time.
Okay, next time. Thank you. And yeah, I apologize that I was not able to be with you guys, but yeah, congratulations for the 30 years.
Thank you.
Okay, bye-bye.
Easy to speak after such nice lectures, but I will do my best. Thank you for this opportunity to share the Amparos journey with you. This journey started 10 years ago when Galvis opening the door to CEPIT NeuroMat for me. I am very grateful for this opportunity. Now, we are reaching the end of this journey within CEPIT NeuroMat, but I hope that this journey can continue within CEPIX NeuroMat. This journey is not only about Parkinson's disease. This journey is about how the brain controls multitasking, how aging process and neurodegenerative disease lead to progressive loss of this capacity, how biopsychosocial factors shape the progression of lack in the functioning, and why this complex process require new mathematical approaches to be fully understood. Then, my goal today is to show how a relevant clinical question leads us from behavior studies to brain image studies to epidemiological studies, innovative interventions, and what is the connection between these studies and the stochastic model, computational science, and artificial intelligence. Why Parkinson's disease? Parkinson's disease is a progressive, incurable, multisystem neurodegenerative disorders. Unfortunately, the neurodegenerative progress starts at least 10 years before the clinical diagnostic is possible. Then, to identify early signs of this disease can open new therapeutic windows. The studies on Parkinson's disease have two important impacts. The first one, the social impact. Parkinson's disease is the second most prevalent neurodegenerative disorders after Alzheimer's disease, but it is the fastest growing among major neurologic disorders with high direct and indirect costs. Then, increase our knowledge on Parkinson's disease can help to improve the quality of life for people, for a million of people living with this health condition, and decrease the healthcare costs. The second impact is the scientific impact, because Parkinson's disease is a very useful model to understand the integration between cognition and action, particularly how the brain resources are managed during multitasking, everyday multitasking. And the knowledge obtained with this kind of studies can be generalized for other populations. And what is the relation between bradykinesia and automaticity? Although Parkinson's disease usually is associated with tremor, bradykinesia is the motor sign that defines the diagnosis. Bradykinesia is a disruption in the ability to select, scale, initiate, and sustain well-learned motor programs, leading to lack to failure of automatic motor control. It is consequence of excessive inhibition of thalamocortical motor circuits and reduced activation of motor cortical motor areas due to dopaminergic loss. Then, at the beginning of this journey, our first idea was that if implicit learning in the goalkeeper game reflects automatic control mechanisms, then performance in the game could predict how well a person manages gait under cognitive demanding condition under dual task. Here, our first results from our first study using goalkeeper game with 74 people with Parkinson's disease. The main results showed that goalkeeper game is a better predictor of complex gait performance in comparison to a gold standard test for evaluation of global cognitive capacity. Then, based on these preliminary results, we can suppose that goalkeeper game could be a free and low-cost, scalable, accessible, engaging measure for automacy, able to detect to early detection of abnormal loss in automaticity. Supposing that goalkeeper game could offer a populational screen, we move to second research question. Where should gait automacy be assessed? In fact, we have a very sophisticated options in terms of motion analysis equipment, but it is not scalable for public health, for example. Then we developed the PANTA solution, Parkinson Affordable Neuromovement Detection and Analysis System. This system uses a low-cost camera and an open source software for movement analysis, the CIVMOB, and we develop a protocol for gait analysis and for postural control analysis. Here, the results from our two first studies showing the results with PANTA for postural control and gait performance. Both studies show that the PANTA system was more sensitive to detect early and subtle alteration in postural control and gait performance than standard clinical tests differentiate the first 3 stages of Parkinson's disease. Then, based on these preliminary results, we can suppose that PANTA could be an option for low-cost, user-friendly, and scalable tool, more sensitive than clinical tests to detect early alterations in the gait and the postural control automaticity. Following this question, we translate the videos in the skeletal key points and use machine learning and deep learning. We develop a model and the main finds show it that the final model achieves perfect separation between Parkinson's disease and health control and between the Hohenhar stages, the main scale for disability level in people with Parkinson's disease. Then, based on these preliminary studies, we can dream of a step care model for Parkinson's disease. Based on the use of goalkeeper game for population level screening, the using of PANTA system in the primary or community health system, and the referral to neurologist and the multidisciplinary team for early diagnosis and early intervention. Then we took a big step, a very important step. We moved from behavior studies to brain imaging studies. We integrate cortical monitoring with gait assessment, postural control assessment, and cognitive assessment using FNARS, a portable system able to detect hemodynamic changes in the brain during a specific task. With particular interest in the dorsolateral prefrontal cortex. Because currently we have two competing interpretations of increased activation in this area during dual task. The first one came from studies showing the association between increased activity with better gait performance. Then, this increased activity could be a compensatory activation, a kind of adaptive recruitment to meet task demands. On the other hand, the second interpretation came from studies showing the inverse correlation. The increased activity in this area associated with worse gait performance under dual task. Then, according to this interpretation, this increased activity could be a reactive overload, a kind of inefficient recruitment due to loss of automaticity. Then, to answer how do longitudinal changes in cortical activation related to motor and cognitive dual task, we develop a longitudinal experimental model, including young adults, healthy older adults, and Parkinson's disease. Currently, we have more than 200 participants in this study. All participants will be evaluated yearly for 5 years, and under 3 conditions: motor single task, cognitive single task, and dual task. Each condition with 10 minutes of duration, 10 repetitions for each condition, using a specific system for gait analysis, and we hope that we can use a force platform for evaluation of postural control and FNARS for cortical monitoring. Here, the results from the first cross-sectional study, including 33 healthy young adults. The main results showed that dual task walking was associated with increased activation in left frontal motor area, not only in dorsolateral prefrontal, but supplementary motor area and the premotor cortex. But we found only a significant correlation between activation in the left dorsolateral prefrontal cortex with gait performance. According to these results supports the role of cortical compensation to preserve gait performance at least healthy young adults. In parallel with these studies on automaticity, we have been working to understand how people living with Parkinson's disease are, where they live, and how social and clinical factors shape their disability. In Brazil, we have a lack of this kind of study. Then this kind of study can help us to understand the Parkinson's disease in real world. To increase our national coverage, we building a national Parkinson network, Rede Amparo. Within this network, we create nationwide online studies, including people with Parkinson's disease from all Brazilian regions. And we are working to building a multidimensional database. Here, the results from cross-sectional studies. The first one showing the showing that the functional declines extends far beyond the motor symptoms. The second showing that age at the disease onset shapes distinct disability trajectories. And this showing that the motor disability and depression predict sexual health in men living with Parkinson's disease, while sleep quality and the couple relationship quality predict sexual health in human with PD. Here, the first results from our longitudinal studies. The first one showing that functional progression does not follow disease age linearly. And the second one, very interesting, territorial vulnerability was the strongest predictor for adoption of regular practice of exercise. Then our next step was to move from diagnostic to intervention. Currently, we have 2 ongoing clinical trials. The first one to investigate the effects of remote mental practice to decrease freezing of cage. This trial asks whether mental practice can modulate the neural mechanisms associated with loss of automaticity. Then the primary outcome is the cortical reorganization after the intervention using FNRs. The second has as main purpose to develop a new app for to promote regular practice of exercise for people with Parkinson's disease. In fact, exercise is increasingly viewed as a potential disease-modifying intervention, but adherence remains one of the greatest challenges in Parkinson's disease. This project has 3 phases. The first one to develop the app, the second one to test its usability, safety, and satisfaction, and the last one, a multi-centric clinical trial to compare the results from new app and printed exercise manual. As primary outcome, we have exercise adherence and several other measures as the secondary outcome. But beyond this clinical trial, the app will collect several important information, such as motor performance, cognitive performance, perceptual performance, severity of symptoms, mood, social support. And we believe that using artificial intelligence, we will be able to identify predictor models that will help us develop more efficient, more personalized intervention for each individual. Here, the results from the first step of this study about the preferences of patients for the app, and here the final design principles. Then we hope that will next step will be using the results from FNARS mapping to identify therapeutic target for non-invasive brain stimulation, specifically transmagnetic stimulation. And since the last year, we have expanded this framework in our studies to people with mild cognitive decline and suspect Alzheimer's disease. Because we believe that dual task gate may become a functional biomarker for neurodegenerative disease phenotyping. Okay, why this is a NeuroMat problem? We believe that the progressive automaticity loss is not a deterministic or random process. It depends on the history, the context and the randomness. Then, using multimodal inputs, the clinical measures, the brain activity, and the biopsychosocial context, we believe that we will be able to predict the future states and the risk of decline. For example, probability of decline, time to transition, risk stratification, and it will be used to not only for intervention, but to reach a better understanding on this process. The same for the functional progression in Parkinson's disease. Using the multimodal information, we can identify profiles of accelerated disability progression, and it may help us to personalize follow-up and intensity of intervention, and maybe to plan resources allocation and health policies. In the same way, we believe that mathematical models may help us identify who responds to each kind of intervention, at what stage and why. Then we can identify different profiles, the strong response, the limited response for each kind of intervention. We are having a very nice experience with our collaboration with Kelly, using computer science to develop digital research infrastructure, to develop tools for data management and integration. For this kind of multimodal data, it is crucial. and to develop digital tools for remote assessment or support for intervention. Finally, we believe that using artificial intelligence will help us to reach the end of this journey over next 10 years. Thank you very much. It is the current studies in your team. I would like to thank the amazing work that they are do. Thank you. Questions?
Thank you, Maria Elisa, for this very important work you've been doing. My question relates to the etiology of the Parkinson's disease, since you are monitoring patients all over the country. And one thing that surprised us and that we had discussed with Antonio Galvez was the correlation with the pesticides. So my question is, in the dispositives or the applicatives that you've been using, if you have any clue or any information that relates to the etiology of the disease, and if what you think about it.
In fact, we have an important lack of this kind of studies in Brazil. And I think that the main barrier is the pragmatism from neurologists. Because for us to build in a kind of national scenario, we can trust in the diagnosis, to identify the new diagnosis. However, neurologists are very pragmatic about the diagnosis. Only movement disorders specialists can offer a final diagnosis of Parkinson's disease. Then my sincere answer to you is no, we don't have. And I think that this work to change this reality is not easy. I have been working for many years for this, and I confess that the advances were very small.
Very, very interesting in all the work you are doing. My question is, are you planning to do diagnosis of other diseases that are like similar, that neurologists confuse between Parkinson and I don't know what is, but
yes, supranuclear supranuclear palsy is the more important differential diagnosis in these cases. I am an optimist, maybe a idealist. Then we believe that we can reach this in the future. But in fact, currently, the diagnostic is basically the clinical exam, and a lot of people are working to identify prodromal signals to differentiate this disease, but it's not easy because it has two important challenges. The first, sensitivity and specificity. Then, for example, the smell loss is a good predictor for Parkinson's disease, but it's not specific. Several health conditions may cause this kind of dysfunction. Then it's not an easy task, but I am optimistic. Okay? Then I would like to thank you for this session. And now I will move for the next important, very, very important session to thank you for very, very nice operational team. Roque, please.
Thank you, Maria Elisa. Thank you to all speakers from yesterday and today. So I think for now we can finish the scientific part of this event and even the English part of it. Let's move to Portuguese because now it's time to make a tribute, to pay a tribute to our team and also to our leader, deceased leader. Okay, so I'll começar a falar em português agora. I'm sorry for the English speaking people, but yeah, I hope you can understand based on the emotional part of it. So, eu gostaria de convidar o professor Bafa, que é o diretor do CEPID, a professora Cláudia Vargas, que está co-organizando esse evento comigo, para a gente conduzir essa sessão, né, de que está marcado no programa, sessão de homenagem, né, às nossas secretárias. Né. Se o Bafa quiser... Cadê? Temos que chamar as duas, então, Então, é importante... Você quer falar, Bafa?
Pode ser.
Seu chefe, né? Então.
Eu queria chamar a Florência também. Florência, vem aqui.
A Florência também, pelo serviço, claro.
Não, a Florência é a vice.
Tá bom.
Okay.
Talvez pudesse acender essa luz.
Agora.
Florência, aqui do meu lado, Florência.
Fica aqui do lado.
Desse lado para equilibrar, senão fica uma foto feia. Alô? Bem, boa tarde a todos. Nós gostaríamos nesse momento de fazer uma homenagem de reconhecimento ao trabalho de algumas pessoas que foram fundamentais para que esse centro se desenvolvesse como se desenvolveu. Obviamente que o professor Galvez foi uma liderança incontestável, já reconhecemos isso várias vezes, mas sem o pessoal de apoio que ele conseguiu agregar aqui, eu acho que a vida teria sido bastante difícil. Todos sabem que o Galvez nos deixou prematuramente e sem aviso prévio. Então, em algum momento, nós tivemos que nos reunir aqui na sala de cima e decidir como é que a gente iria continuar esse projeto. Que é um projeto, como já foi falado, multidimensional, não só no seu escopo, por envolver áreas muito distintas, com maneiras de pensar diferentes, com demandas diferentes, e, além disso, um desafio que era tentar fazer algo que fosse realmente inovador. Nós tivemos, nessa fase de transição, vários diálogos com a FAPESP, e confesso que foi muito gratificante ontem ouvir o depoimento do professor Luiz, que era a pessoa que estava do lado de lá, e demorou um certo tempo até que eles entendessem o que era o CEPID NeuroMat. Porque normalmente os outros CEPIDs que tem por aí, não estou fazendo nenhuma crítica, mas é um modus operandi, eles eram uma constelação de grupos de pesquisa que se reuniam, conseguiam os recursos e depois seguiam a vida com uma interação muito pequena. E o Galvez fazia questão de que isso não se repetisse aqui. Ele queria que a gente trabalhasse com um objetivo único. Lógico que você podia fazer outras coisas, tinha as outras preocupações, mas enquanto estivesse aqui no CEPID, a gente tinha que ter integração, a gente tinha que ter contatos. Acho que isso nesses 2 dias ficou claro que existem. Não são coisas simples, não são coisas que ocorrem do dia para a noite, ou da noite para o dia, demora um certo tempo, mas a gente vai convergindo. No meu caso, por exemplo, nós começamos com instrumentação, e hoje a gente tem 3 projetos que, de alguma maneira, está testando essa hipótese de estaticidade, de ver como é que o cérebro responde, etc., sem prejuízo de buscar outras cooperações com outras áreas, com outras aplicações. Bom, isso posto, vocês podem ver o impacto que isso tem na gestão, né? A gente tem que comprar coisas para um grupo, coisas para o outro, para outro. As demandas são múltiplas, tá certo? Se a secretária, que é a Lourdes, que está sentada no fundo, tivesse só cuidar de matemático, ela já é PhD nisso, tá certo? Fez a vida inteira. Mas aí vem um físico, aí vem uma fisioterapeuta, né? Aí vem um engenheiro. São demandas diferentes, né? Tem que comprar um negócio, tá certo? Eu lembro que a Maria Lúcia, uma vez, nós tínhamos que montar a base dos braços robóticos. E aí tinha que comprar uns vergalhões de alumínio. Ela conseguiu um preço melhor do que eu consegui. Não sei como, mas isso mostra, né, que sem a ajuda da Marilúcia, né, e da Lourdes, tá certo? Ou da Lourdes e da Marilúcia, uma dobradinha ali, né, a gente não conseguia chegar onde chegou. E fizemos isso muito bem, fizemos isso com cuidado, afinal a gente está lidando com recursos públicos, né? E, bom, então, aquela conversa naquela salinha ali que decidiu que eu iria ser, tá certo, o condutor do CEPID, não seria possível sem a ajuda delas. Depois, logo em seguida, a Florência também se juntou ao time na coordenação. Então, eu queria aqui agradecer, né, de público, a colaboração que foi inestimável da Lourdes, da Maria Lúcia, tá certo? Tivemos que fazer ajustes também. A gente jamais queria que a Maria Lúcia tivesse saído quando saiu. A gente gostaria de estar terminando junto agora, mas infelizmente as condições, né? Estava comentando aqui com o Pantelis. Hoje em dia o pessoal valoriza mais a forma do que o conteúdo, né? O controle do que o resultado. Devia ser o contrário, mas é a nossa sociedade, não vamos discutir isso agora, que nós não vamos terminar. Então, eu acho que esse momento é para isso. Lourdes e Marilúcia, é para agradecer imensamente a dedicação que vocês tiveram, as horas que vocês tiveram. Muitas vezes a gente trabalha 24/7, né? Ainda esse fim de semana tivemos problemas, aí a Lourdes teve que ir no sábado se acionar à noite e tal. E, enfim, agradecer imensamente. Acho que vocês passaram para as pessoas que estão aqui um exemplo que tem seguido. E, bom, a vida segue, a gente vai continuar com outras coisas e espero vê-las em outras ocasiões, mas muito obrigado.
Podem vir aqui, Marilúcia e Lourdes.
Tá bom.
Não sei.
Obrigada.
Como uma forma bastante singela de marcar esse evento, a gente fez uma placa que entregou agora para a Marilúcia e para Lourdes. Não sei se elas querem ler.
Obrigada, professor. Eu tenho que falar?
Fala.
Bem, eu queria agradecer. Foi muito bom trabalhar. Foi dos 40 anos de profissão de secretária, os últimos 13 foram bem mais assim corridos, mas eu aprendi muito. Tive a oportunidade de trabalhar com o professor Galves, que a gente ia almoçar e ele corrigia meu inglês, que não é nada, né? mas me punha uma fria danada para levar professores internacionais para almoçar. E eu, sem saber falar direito, ficava tentando ver se eu conhecia alguma palavra para poder dar continuidade no assunto, mas levava o pessoal e acho que ninguém reclamou até hoje. Então, eu agradeço, foi um trabalho que me fazia bem feliz. Eu agradeço ao professor, aos professores que, depois do professor, continuou comigo. E muito obrigada a todos pela confiança. Obrigada.
Quer falar alguma coisa?
Eu vou ler aqui a placa. A Lourdes Vaz da Silva Neto, em reconhecimento à sua dedicação, competência e incansável apoio ao CEPID NeuroMat ao longo de sua trajetória. Seu trabalho, sempre realizado com profissionalismo, generosidade e compromisso, foi fundamental para o desenvolvimento das atividades científicas, acadêmicas e humanas do Centro. Nossa profunda gratidão por sua contribuição para a construção da história do NeuroMat. Eu agradeço e só queria concluir uma coisa. Eu acredito o seguinte, que a gente conseguiu chegar até aqui com sucesso, não só do nosso trabalho, como dos meninos lá atrás, o Arthur e o Matheus, além de toda a equipe do IME, que a gente pede socorro, deu algum problema, saem correndo, vem aqui e nos ajuda. Mas agradeço pela oportunidade. Eu achei muito bom vir para cá quando o Gauss me convidou e aprendi muita coisa. Muito obrigada.
Isso, acho que a gente tem que também agradecer Arthur e Matheus, que estão aqui desde faz, acho que 1 ano e meio com a gente e que têm dado todo o apoio também para a realização do workshop. E, bom, como o Lourdes falou, também tem muita gente por trás no IME trabalhando para que aqui as coisas funcionem. Então, obrigada.
Alguém quer fazer uso da palavra? Algum outro participante?
Bom, eu sou a minha unidade de origem, a Faculdade de Medicina, né? E eu estou nela desde 88, primeiro como aluna e depois como docente. E olha, eu queria dar meu testemunho aqui, porque quando eu comecei a trabalhar aqui no NeuroMat e conheci a Lourdes e a Mariluce, eu não sabia que na USP poderiam haver 2 pessoas tão engajadas, tão prontas a fazer tarefas que não eram delas, tão pronta a nos auxiliar. Então, em toda essa minha trajetória, tanto a Faculdade de Medicina, até hoje a Lourdes se oferece para fazer algumas coisas que eu pego e fico assim, pô, mas isso é obrigação minha, Lourdes não precisa fazer isto, e ela sempre muito gentil e muito comprometida e muito disponível, assim como a Mariluce, que também fazem muito mais do que o combinado. Então, meu agradecimento é esse, é porque vocês, em toda a interação comigo, fizeram muito mais do que o combinado. Muito obrigada.
Ok, então agora...
É isso aí. Bom, agora, então, essa parte do evento foi encerrada. Agora vamos para a última parte, que é o filme feito em homenagem ao fundador do CEPID NeuroMat, né, nosso mentor, o professor Antônio Galves, João Pechansky, que é um dos diretores, um dos produtores do filme, chamado aqui para...
Português ou inglês?
Não, pode ser em português, porque o vídeo está em português. Né?
Eu pus legenda.
Você pôs legenda? Então você pode dizer isso. Né?
Tá. Olá, o filme é 1 hora, vou falar 5 minutinhos. Tá, então, boa tarde, para quem não me conhece, eu sou João Pechansky, atuo na difusão do CEPID NeuroMat. Então, antes da gente apresentar o filme, é um filme de 1 hora, então vocês aguentem um pouquinho. Eu vou só dar um pouco de contexto sobre a área de difusão e o trabalho, como é que ele acontece. Desde o início, a FAPESP, quando ela imaginou esse conceito institucional, essa visão que é um CEPID, lhe deu uma importância muito grande à área de difusão. CEPID é Centro de Pesquisa, Inovação e Difusão. E, de certo modo, obrigou, mais do que recomendou, que esses centros que nasciam tivessem dentro dele uma área voltada à ampliação e melhoria, qualificação da cultura científica. E dentro do CEPID, a gente fez isso com enorme investimento, né? Fizemos um conjunto de iniciativas e hoje eu vou apresentar para vocês uma delas. É uma das últimas, que é um filme em homenagem ao Antônio Galvez. Foi o líder inicial desse projeto. E a ideia do modo com o qual nós trabalhamos foi trazer para dentro do CEPID bolsistas de jornalismo científico, uma modalidade de bolsa da FAPESP. Foram 29 bolsistas do nível pós-graduação, 17 no nível de graduação dentro do CEPID, ou seja, nós basicamente criamos um enorme consórcio de comunicação, difusão dentro do CEPID, da área que é das a comunicação multimediática, ciência da informação, também passando por jornalismo fotográfico, jornalismo textual, podcast. E a ideia dessas bolsas é que todo projeto ele teria que fazer duas realizações, uma delas mais operacional. Todo bolsista realizou o seu próprio produto, e trabalhar com produto, como um filme, vocês vão ver, é uma outra dimensão de trabalho que envolve, por um lado, a criação de um roteiro, antes mesmo do roteiro, uma escaleta, uma visão do processo, mas sempre num enfoque de experimento. E essa noção de experimento conecta com essa segunda parte, que era todo bolsista de jornalismo científico, que é uma área mais operacional, a gente chama de ciência social aplicada, também realizava um projeto de pesquisa. E a base da pesquisa desse filme, que vocês vão ver, é a Tani Sugano, que é a estudante que realizou esse trabalho, foi, por um lado, entender como é que, numa revisão de escopo, como é que a biografia científica tinha sido imaginada, como é que você contava a obra de pessoas a partir daquilo que elas tinham produzido. Então, muitas de vocês devem ter visto filmes sobre grandes cientistas. Então, o Einstein, geralmente são homens, enfim, a gente só achou dentro da literatura realmente biografia científica sobre a Marie Curie, no universo, enfim, dessas grandes personalidades científicas. E, por outro lado, e conecta com isso que eu acabei de falar, como é que a gente contaria a história deste líder sem fazer aquilo que no documentário é o caminho mais fácil na estrutura, que a gente chama da narrativa do herói, em que você, principalmente no ambiente do filme obituário, você cria uma narrativa que faz essa pessoa se tornar, enfim, monumental. E como é que a gente traria elementos mais sensíveis e mais humanos mesmo dentro dessa construção, que também era uma homenagem. Então, deixo vocês aqui agora com o filme em homenagem ao Galvez, que foi realizado pela Tani. Eu estive na função de supervisor e também produtor executivo. Essa é uma obra que, infelizmente, ainda está embargada, ou seja, eu não posso mostrá-la porque nós estamos concorrendo num festival de filme científico. Chegamos, por enquanto, na fase de final. Somos um dos 200 filmes que será avaliado até setembro, e depois disso espero realmente popularizar esse documento, porque nós temos um compromisso bem importante com a difusão livre. Just in English a little bit. The movie has some rough subtitles, just for our non-Portuguese speakers. They are rough, as I said, which means they were done like just a couple of weeks ago, so I beg your pardon for any misspelling or typo. So, thank you very much. Pode passar, por favor. Eu que passo aqui?